Okay, am I audible to all of you?
Yes, ma'am. Yes, ma'am.
Yes, ma'am.
And the slides, they are also clearly visible, right?
Yes, ma'am. Yes, ma'am.
Okay, so let's start with a few topics from the leftover class. Okay. So the next topic to be discussed is regarding the penetrance and expressivity. Okay. So sometimes what happens, the gene or the genotype, it gives a variable expression in a population. Okay. Like as you can see here in the first case, so there are 10 individual, but only 1, 2, 3, 4, 5 of them are actually showing the phenotype associated with the genotype. The rest five, they are not showing the genotype, the phenotype that is associated with the particular genotype. So in this case, we can say the trait is not 100, the trait does not have a 100% penetrance level, or we can say it has a variable penetrance. Now, sometimes the genotypic expression may be variable within a population. Means, in the second case, if you observe, every individual is expressing the genotype, the phenotype associated with the particular genotype. Because you can see slight color variations in all the members of the population. But look at the intensity of the color. It is very different from one individual to another individual. So here we can say the trait has a variable expressivity. So penetrance means it actually measures what percentage of the members in a population are exhibiting the phenotype associated with the genotype. Like for example, let's say if in a population more means 10% they are having a capital T small t phenotype genotype. So all of them should be tall, but it might be possible that only few of them are actually will be tall and rest may not be as tall as expected. So that is what is called as penetrance, like how much the genotype or the phenotype has been penetrated into the population. Then expressivity means whatever members of the population are expressing the character, what is the intensity or what is the variability of the character, whether all of them are expressing the same character or there is a difference in the degree of expression of the character. That is what is called expressivity. So the last picture you can say it is exhibiting variable penetrance as well as variable expressivity, because see this individual and this individual, they are not expressing the character actually. So here the trait is not 100% penetrant, or we can say it is exhibiting variable penetrance. Moreover, whatever individuals are expressing the trait, there is a variation or there is a variation in the degree of the expression. See, this is very dark, but this is comparatively lighter, this is more light. Okay, so whatever characters are expressed by the rest of the member, the characters are not uniform. Some are exhibiting more amount of character or more degree of the character in respect to other. This is what is called as variation in the expressivity. So next concept is called phenocopy. So phenocopy means sometimes what can happen, there could be a phenotype which is not actually due to a genotype, but rather due to a variation in the environmental condition. Okay, but the phenotype will be like a copy of the actual genotype that exists. For example, here you can say like Drosophila, the fruit fly, if we take into example, the fruit fly sometimes can have a defective kind of wing structure. Okay, so the defective wing actually occurs due to a mutation in the gene that is present in the fruit fly. But sometimes what happens, if there is a wild type gene or there is a normal fly, but if the fly is treated with a heat, means if it is raised in a high temperature or something, but genotypically it is normal, but if it is subjected to a variable environmental condition like extreme high temperature, then whatever phenotype will occur, it will be same as that of a genetically mutant phenotype. So this kind of phenomenon is called as phenocopy. So phenocopy, simply you can say the copy of a or copying of genotype of, sorry, copying the phenotype of a genotype. Like if there is an actual mutant, the mutant will express a kind of phenotype. Okay. So for example, if there is a fruit fly, it has some mutant gene, then it will have this kind of that tetraptera condition. But sometimes a wild type fly with a normal gene is, if also it will be given to a, or it will be subjected to a different type of environmental condition or to a high temperature condition, then instead of having a normal gene, the phenotype will be like that of a mutant phenotype. So this phenomenon is called as a phenocopy. Okay. So this can also happen in some of the animals. Sometimes it can be called as the environmental effect on a normal genotype. Then comes the concept
Ma'am,
of,
ma'am,
yes.
uh, the phenotype is equal to epigenetics.
No. Epigenetic is something very different. Okay. Epigenetic we will discuss today. So wait till the end of the presentation, you will get to know. Phenotype, I think I already explained you yesterday also. Simply whatever character you are observing, that is called as a phenotype. Okay. Epigenetic is something
And
different.
phenocopy. Phenocopy is
No, no,
similar
no,
to
no.
epigenetic.
That is no. Phenocopy is not same. I explained already. Phenocopy means sometimes, sometimes the phenotype is actually determined by the genotype. Okay. So, okay, let me give you another example. Like, for example, if the plant is having a genotype like this, capital T, small t. See, the plant is having a genotype like capital T, capital T. So just tell me what kind of a character you are expecting from this plant.
Tall.
Tall. Okay. Okay. Now tell me if a plant is having a genotype like this, small t, small t. So what kind of a character you are expecting from this plant?
Dwarf.
Dwarf. Okay. Now, if you grow this tall plant in an area where there is no nutrition, no sunlight, nothing is there. So, although it is having a tall genotype to express a tall phenotype, still the plant will appear dwarf. So genetically it will be normal, but phenotypically it will be dark dwarf because it does not have adequate nutrition, it does not have sunlight, or it does not have the factor that will help it to attain the maximum height. So in this case, this dwarf phenotype is not it exactly how this plant should be, but actually the plant will be having a genotype like this. But instead of having a normal genotype, if it will express a phenotype of another genotype, that condition we are calling as phenocopy, which is simply because of a environmental factor or environmental effect. Am I clear now?
Yes,
That is
ma'am.
what
In
is
the
called as a phenocopy.
epigenetics,
No, epigenetics is something very different. Okay, epigenetic is something different. So I will explain it that later.
okay, ma'am.
This is due to environmental effect. Then coming to the concept of gene. So if we think of like exactly what we mean by gene, so gene can be expressed as three different units on the basis of its function. So gene could be treated as a unit of transmission that we call as cistron. Okay. Or gene can be regarded as a unit of recombination, that is recon. Or gene can also be regarded as a unit of mutation or muton. So first, if we look from genetics point of view, mostly we will be sticking to cistron because what we are thinking gene as like this is a this is a factor which is actually inherited from one generation to another generation, and it actually controls a particular trait or phenotype. Similarly, from molecular point of view, if you observe, the genes, they are a segment of DNA which is capable of undergoing a process of recombination. So this could be also treated as a unit of recombination. Now, from molecular point of view again, the gene, it is a sequence of DNA where the sequence change or the change in any kind of structural formation of the DNA could lead to a alteration in the phenotypic character. So these are the modern concept of gene, where we are thinking gene as a unit of transmission, a unit of recombination, as well as a unit of mutation. So are there any queries from anyone? Okay. So there comes again the concept of one gene and one polypeptide, or one gene, one enzyme hypothesis, given by Biddle. So as per the concept one gene, one enzyme hypothesis, a particular gene encodes a particular enzyme that can
Excuse me,
control
ma'am.
a
I
metabolic
have
path.
one doubt, ma'am.
Yes,
Why this process happen and what is the importance of this type for gene 1, 2, 3? Why this, what the role of this process?
no, no, this is just an example to show that one gene actually encodes a single polypeptide. The single polypeptide can form a particular enzyme which will be controlling a single metabolic pathway. So if multiple genes are involved or multiple enzymes are involved in a single pathway, even if one gene is interrupted, then the entire metabolic pathway can be disrupted. So the whole concept of this diagram is to predict that, or to explain that a single polypeptide is encoded by a single gene. That is what is called as one gene, one polypeptide, or one gene, one enzyme hypothesis. Okay. So
What is the
next
role of
comes,
a gene in a human body?
can you please keep your questions towards the end of the discussion, because if I keep on discussing this, we are getting delayed in the syllabus actually, and the other people, they will be having problem with that. So please wait till the end of the presentation. Then whatever questions you have, you can ask them. Okay, or else we won't be able to finish the syllabus.
Okay, ma'am. Okay. Please continue.
So next comes the concept of gene, like we discussed here, the concept of cistron. So one cistron actually represents a particular gene or a particular sequence that will be actually encoding a single polypeptide chain. Okay. So this was actually proved in an experiment called as the T4 bacteriophage experiment, which was conducted by Seymour Benger. So what he did, he actually took two different type of bacteriophage. Okay, so bacteriophages are viruses actually that that actually engulf or kill the bacteria. The bacteriophages, they actually consume or they kill the bacteria. So here you can observe that he has taken two different mutant forms of a bacteriophage. They are depicted by two different colors, as you can see here. One is red and one is blue. So these mutant form of the bacteriophage, they have a particular gene called as the R2 loci. Okay, so what is the function of this R2? Now this R2 loci actually kills the bacteria and forms colonies like this. Okay, so as you can see on the plate, there are different type of bacterial colonies are there. So these R2 mutant or these R2 bacteriophage, they create colony, they kill the bacteria and create colonies like this. Now, this mutant bacteria, bacteriophage, they can actually infect a bacteria, the bacteria that is called as E. coli, E. coli K. Okay, so the mutant form of the bacteriophage, they can kill the E. coli, the bacteria E. coli K. But there is another type of bacteria which is called as E. coli K. lambda. Okay, so the mutant form of the bacteriophages, they cannot infect and they cannot kill this E. coli K12 lambda. Now, what he observed when he co-infected two different type of mutant bacteriophages into a single bacteria, so all of them could lyse and kill the bacteria, forming this kind of colonies. So this is absolutely normal. Now, what he observed, few of the bacteria which actually in original these two bacteriophages, they cannot kill the bacteria. Okay, the bacteria E. coli K12 lambda. But what he observed, when he co-infected them into a single bacteria, few colonies developed. So what could be the possible explanation? The two bacteriophage who separately cannot kill the E. coli K12 lambda, how come upon co-infection they could lyse this kind of bacteria? Now because it might be possible that the mutations that are present in the bacteriophages, they might be present on two separate segments of gene. Okay, so this test is actually, he called this test as cis-trans complementation test. So if by chance the mutations are present on two different genes, like as you can see in the first case, so here the mutation is in the first bacteria, the mutation is in the gene 1. In the second bacteria, the mutation is in the gene 2. So if they undergo recombination, then what you will get? You will get, so these two parts will be exchanged. So you will have two normal copies of gene 1 and gene 2. So this is exactly same like that of a wild type bacteriophage. So this wild type bacteriophage can lyse the K12 lambda. But by any chance, if both the mutations are on the same gene, that you can observe in the second case. Okay, so if both the mutations are the same gene, then it will behave as a mutant because there will be no complementation. So this is what is called as the cis-trans complementation in bacteriophage. So which indicates actually, if the mutation is in transposition, then they can be complemented and you can get a wild type character like this. But so this is actually called a trans complementation because the mutations are in the transposition. Trans means two separate DNA strands. Now, if the mutations are on the same gene, which is called as a cis complementation. Okay, so this in this case the mutations cannot be complemented. So you will never get a wild type phenotype in this condition. So this test, which I told you that known as the cis-trans complementation test, was explained and was established by Seymour Benger in T4 bacteriophage. So this actually gives the concept of a gene. So what is exactly the outcome of this experiment? Now, if there is mutation in two separate genes, they can be complemented, they can complement each other. But if the mutations occur within the same gene, which is actually a cis mutation, so there won't be any kind of complementation, and this unit of gene is called as a cistron. So if there is a mutation within the same cistron, there will be no complementation. But very rarely the cistrons, they can undergo recombination, forming they can undergo recombination where you can get a wild type copy of the gene and a double mutant copy, from which you can actually calculate what is the distance between the mutation site. So this cis-trans complementation test is also known as the fine structure analysis of gene, which actually gives this, that a set of gene, or we can say a sequence of gene where the mutations cannot be complemented to each other should be regarded as a single cistron. Okay. So I hope you understood, all of you understood this part. Please go through your textbook and if you face any doubt, you can ask me tomorrow. Okay. So this is nothing but a simple test. Here, if you focus on this part only, you can understand. So when the mutations are in two different
Madam,
genes,
madam, don't mind please. Actually, आमे टिके बुझी पारुनु. तो बुझी पारथा तो आपण को क्वेश्चन काई पचारथांदो. आमे तो एकरे क्लास तो करबा को जाई पारुनु. आपण पढिला बेले जो पेज रे डाउट आसीबो, मैडम देखंतु आपण जो पेज रे पढौचंदी सेही पेज रे डाउट आसला, सेठी न पचारिले आपण लास्ट जो रखिबे ता भितर आमे पांचटा स्लाइड रे पांचटा डाउट थिबो, केमिति पचारिबो एका संगे सब क्वेश्चन कोड गोटे मिस हे जाउची.
देखंतु मो कहिबार ये
आ गोटे मैडम खराब भाबंतुनी मैडम प्लीज, आपण पूर्व क्लास जो स्लाइड गुडा रहि जाईची
ना ना मोर प्रॉब्लम टा मु कहि दिए देखो मते लिमिटेड टाइम मिली आपण को भास्ट देखो जो 4 दिन भीतर मते चारिटा को
मैडम
ब्लॉक
खराब
कंप्लीट
भावतु ना एक्चुअली मैडम पूर्व क्लास रे जो सार पढ़ौथिले ना आमे
आज्ञा
किछ से कथा कहू ना आपण टिके माइंड करिवेनी से पूर्व क्लास रे सार को आमे जोड पचारथुलु सार सबुडी सेठी बुझेई दउथिले आज्ञा
ना ना मु बुझिली मैडम आपण कथा मु बुझेबा पई मना करूनी किंतु बाकी भी स्टूडेंट
ଆଉ
माने
ଯୋଉ ପୂର୍ବ
ऑब्जेक्शन
କ୍ଲାସ
करि
ର
परिबे
ସ୍ଲାଇଡ
जे
ଗୁଡାକ ସବୁ ରହିଗଲା ନାଇଁ ତ ମ୍ୟାଡାମ କେହି ତ କିଛି କହିନାହାନ୍ତି ଆମେ ଏମିତି ପଚାରୁଥିଲୁ ପୂର୍ବ କ୍ଲାସ ରେ ସାର୍ ଙ୍କୁ ଯିଏ ଯାହାର ଡାଉଟ ଥିଲା ପଚାରୁଥିଲେ
बुझिली मैडम सुनंतु किंतु काली देखंतु मोर ऑलरेडी ब्लॉक 1 रो मु काली पूरा भी कंप्लीट करि पानी आज भी मते बहुत सारा जिंस आगो को एक्सप्लेन करिवार
ମ୍ୟାଡାम
अछि
ଯଦି ଆମେ ଛୁଆଙ୍କର ଡାଉଟ ଟା ସଲ୍ଭ କରିପାରିବାନି କିନ୍ତୁ କୋର୍ସ ସବୁ ସାରିଦବା ସେଇଥିରେ ପିଲାଙ୍କର କଣ କିଛି ଇମ୍ପ୍ରୁଭ ହେଇପାରିବ ମ୍ୟାଡାମ
Madam, may I say something?
हां हां कहि पारन किछि असुविधा नै कोंतु
जो मानकर डाउट अछि से माने लास्ट रे न खाता रे लिखिकी रखंतु जे एई जगह रे हमर एई डाउट टा अछि मैम को प्रति टाइम रे जदी आपण एमती माने पचारिबाटा भूल कथा नुहे किंतु आपण जदी प्रति टाइम रे पचारिबे तो मैडम जहा पढेइबा पई आछन सेटा ही पढेइ परिबेनी
एक्जाक्टली एक्चुअली मो प्रॉब्लम टा मु से ही कहया चाहूची मो मना करून जे आपण क्वेश्चन करंतुनी बोली क्वेश्चन जदी न करिवे बुझिबे केमती
आपण आपण खाता रखिकी माने खाता रखिकी
ठीक
बसिना
अछि ठीक अछि लास्ट को मैडम कहिले तो आपण पचारिबा पई हम लास्ट को पचारिबो आगे
हाँ आपण टिके देखंतु लास्ट को हाफ एन आवर रखिबा जोथरे आपणकर आ किछि मो डिस्कशन करिबिनी जहाँ भी जहाँ क्वेरीज अच्छे सेतिक बेले सॉल्व करिबा कारण एमती जदी आपण सब स्लाइड रे पचारि पचारि चलिबे ताले प्रॉब्लम अछि
ओके
कि हम
मैडम ठीक
कवर
अछि ठीक
ओके ठीक अछि तो फाइन सो कोड गला ओके सॉरी ओके सो जस्ट ये माने समरी मु कहि दउची एक्चुअली एथरे अच्छा के नॉन ओडिया पिला अछंती कि ना मु जदि कंप्लीटली ओडिया रे एक्सप्लेन करे किछि प्रॉब्लम होबो कि
No, ma'am. Sorry, ma'am, but I will not be able to understand a single thing.
Okay, okay, fine. So that is also another problem. Okay, see, in between I will be means telling in Odia. So if you face any problem, just inform me. Okay, like I will be going bilingually, but in order to make them understand a bit faster, it will be easy if I try to explain it in Odia. Okay, so let me know if you face any difficulty within that. सो देखन मु ऑलरेडी एटा तो एक्सप्लेन करि सारछि आ थरे कहि दउछि देखंतु हमरो ए जो टेस्ट थिला कॉम्प्लीमेंटेशन टेस्ट सी स्ट्रेस कॉम्प्लीमेंटेशन टेस्ट यार एक्चुअली आउटकम होछि कोन ना हमरो जदी आमे गोटे जीन को गोटे सिस्ट्रन को गोटे यूनिट धरुचे सो जदी सेम सिस्ट्रन भितरे म्यूटेशंस होछि जेमति तमे एई जगह रे देखि परिबो देयर विल बी नो कॉम्प्लीमेंटेशन सो सेठो हमर एक्चुअली सिस्ट्रन रो कांसेप्ट आछि कि if there is a sequence of DNA where there is mutation and that mutation cannot be complemented, this is called as a unit of gene which we are calling a cistron. So jete samay jae tomar dekho jodi bahare dita different gene re tomar mutations achi tahale kon hobo na recombination heiki recombination hele kon hobo na e jo normal gene ta एपट सेगमेंट को पळेला आउ ए जो डिफेक्टिव जीन टा एपट को पळे आसला तो तमरो जो बैक्टीरियोफेज तयारी हबो हाफ ऑफ द बैक्टीरियोफेज विल हैव माने समस्त नॉर्मल जीन रहिबो आउ हाफ ऑफ द बैक्टीरियोफेज रे कोन हबो केवल म्यूटेंट जीन टा रहि जिवो सो एई जगह रे कोन ना तमरो कॉम्प्लीमेंटेशन हे गला सो जदियो इंडिविजुअली दीटा को बैक्टीरियोफेज रे म्यूटेशंस थिला बट बिकॉज ऑफ रिकॉम्बिनेशन they are now mane few of or 50% of them will behave as a wild type so because of wild type behavior they can now lyse the K12 lambda as well as K12b so सेटा ही मु कहया चाहूँ कि that is the cistron that is what is called as a cistron जो यूनिट भितरे जेमति देखो एठी तमर कोन दीटा को म्यूटेशन सेम जीन रे अछि so here the complementation cannot occur because both the mutation belong to the same gene or they belong to the same cistron yeah. Okay, fine. Then coming to the actually next we have the concept of linkage and crossing over. So Mendelism रे जदि आमे देखिबा, so Mendelism रे कोन थिला ना कोनोसी प्रकार linkage आमे देखिया को पाइ नथिले, कारण all the genes that were encoding the character, actually they were present in separate chromosomes. But sometimes what can happen, the character that encode or two separate gene that control the two different character might be located on the same chromosome. So in that case, the characters might appear linked. So linked कहले कोन ना जेतेबेले तमरो जेकोनोसी गोटे क्रोमोसोम उपरे जदि दुईटा जीन अछि and they are controlling the same character, in that condition the character means if you cross two different individuals with the same character that are present on a single chromosome and the distance between the genes is comparatively less, then what you will observe, you will observe that the parental type of combination will be more. So this is what is called as linkage. So linkage we can define as the tendency of two genes to move together during meiosis due to means precise location or due to near location on the same chromosome. So jetale tamara gene dita bitare when the distance between the genes is more, in that case the possibility of crossing over will be more. So here as you can observe here, this is the diagram of the chromosome. So now if we think, so here if you observe, this is let's say here there is a location of gene is here and the another gene is located here. So since both the genes are in a measurable distance, the chromosome or the chromatid parts can be easily exchanged and you can develop a recombinant character here. Means this will have a, let's say if this is having a capital A and this gene is, this segment is having a capital B gene and this is having a small a, this is having a small b. So after the recombination, what you can observe, the capital A will have the small b alongside it, the small a will have the capital B alongside this. This is what is called as a recombination character, okay, or the recombinant. So the recombination is only possible when there is a distance between the genes or the genes are means located in a measurable distance. But imagine if both the genes are located very close to each other like this. This is the A, this is B, then this is A, this is B. Okay. So if they are very close to each other, it is difficult to carry out a crossing over between them. So what you will observe, in all the progeny or all the gametes will have always capital A with capital B and small a with small b. Or in other terms, if we take the example of the yellow and round condition, if we say, so if the yellow seed will always be round in shape and the wrinkle seed will always be green in shape. So you will not have any new character over here. So this concept is called as your linkage. Now the linkage could be of two different type, it could be 100% linkage or complete linkage. So complete linkage means the genes are very close to each other and they will never be separated from each other. And in incomplete linkage, as we saw in this case, the genes can undergo recombination where you can have a round gene with wrinkles, a yellow gene with wrinkle shape and a green gene with round shape. Now, another thing here you should keep in mind that the maximum frequency of recombination could be 50%. So, if which is actually occurring in case of your Mendelism, there you can observe that the maximum means the recombinant, they can occur in 50%, which is a case of independent assortment. Because if the genes are located 50 map unit apart, so there is very means maximum chance that they will every time they will undergo recombination and the characters will become separated from each other. So you will have all the parental as well as the recombinant characters in equal proportion. So this thing you should keep in mind, what could be the maximum frequency of recombination. This is always 50%. Okay. So let's take an example where it will be easier for you to understand. So here you can Okay.
Ma'am,
Okay,
may
so
I ask a question?
yes, yes, you can ask.
Ma'am, the recombination will happen between X and Y chromosome?
No, no, no. Recombination will always occur only between homologous chromosomes. Okay. So if means they should be having the exactly the same set of genes. So only with different allelic means combinations, the recombination will occur. The recombination between non-homologous chromosomes is called a translocation. That is actually a mutation. That is actually means bad thing for the cell. It's not a good thing. Okay. So that is a mutation actually. Translocation mutation. So always recombination means it has to occur between the homologous chromosomes, because only then they will have exactly the same set of gene, but the allelic combination might be different. So that is how you will be having different set of characters or different set of phenotypes.
Between
So
XX chromosome, there will be the recombination.
yes, between two X chromosomes, there will be recombination. Okay. So maybe
Okay, ma'am.
one X chromosome will have the normal gene and the other X chromosome will have the hemophilia gene. One X chromosome might have the colorblind gene, the other might have the normal gene. So accordingly, they can undergo recombination and you can have different combinations. Like you can have a normal gene with hemophilia, you can have a means both sickle cell and hemophilia, like that. Okay. So it can occur, but it will not occur between two different chromosomes. So here, as you can observe, the first Drosophila, it is having a long wing with a gray body. Okay. And this is having a vestigial wing with a black body. So this is a wild type character, means this is a means commonly found in the population, and this is a mutant or the recessive character, because the characters are not very good, or means they are not very useful for the organism. So the wild type genes are always presented by VG plus, like the VG stands for the vestigial wing. So vestigial, this is the wild type form. So this is why it is VG plus, and this is B stands for the black body. So since it is having a gray body which is dominant over the black one, it is called as a B plus. So since this is an diploid organism and it is a wild type phenotype it is having, so it will have VG plus, VG plus, B plus, B plus genotype, and it is crossed against a mutant. The mutant is having both the recessive character, vestigial wing and black body. Okay. Now, what kind of gamete you will get from this parent? So you will have VG plus and B plus. Okay. So this is the gamete that you got from the first parent. Now, what kind of gamete you will get from this? Since both of them they are same, you will also get a VG and B combination from this parent. So you will get a hybrid for the F1 generation. Okay. So in hybrid, so what you had? Now VG plus, VG and B plus and B. Okay. So since it is having the wild type copies of each gene, this will give rise to the dominant phenotype, that is long wing and gray body. Okay. Again, this plant, this hybrid fly is crossed with a double recessive mutant. So what kind of a character it is having? Vestigial wing and black body. Now, since this is also recessive character, or this is a recessive genotype, you will get only one type of gamete from this, that is small VG and B. But this hybrid can have 4 different type of gamete. So what could be the possibility? Now maybe there will not be any kind of recombination and the chromosomes will separate like this. So one gamete will have combination VG and B. The other gamete will have combination VG plus and B plus. But if there will be recombination between them, so or crossing over between them, so what combination you will have? This B plus will go here and the B will come back to here. So you will have a combination that is VG and B plus, VG plus with B. So you will have 4 different type of gamete from this. Okay, so you will have 4 different type of gamete from this individual. So that is what? VG plus B plus, which is same as this gamete. Okay. this one. Then you will have another combination VG and B. So these are actually called as the parental type. Then you will also have a new combination because of recombination, that is VG plus B and VG B plus. This part understood, everybody? So when the gametogenesis will take place, either there will be no recombination, the chromosomes will separate like this with the original combinations, or they might undergo crossing over where you will have a combination like this, VG plus B plus, sorry, VG and B plus and VG plus and B, where these segments have been exchanged. So accordingly, you will have 4 different type of progeny in the F2 generation. So what will be the combinations? Now it will be VG plus B plus and V and G from this parent. Okay, this one you will have, or you can have VG and B, VG and B from this parent, and VG and B from this. Okay, or you will have a VG and B plus from this parent, VG and B from this, then VG B plus from this parent, VG and B from this parent. So you have 4 different type of progeny where these two are resembling the parental combination, because in parent what you have, long wing gray body like this, vestigial wing black body like this. But you have 2 new characters, that is vestigial wing with gray body, which is not present in the earlier generation. Similarly, you have also long wing with black body. So these 2 new combinations are called as recombinants. Now, how, if you look into the number of parental combination is much higher in comparison to the recombinants. So why? Now, this is actually dependent on the distance between the genes. The more will be the distance, more frequently the recombination will occur and you can have higher number of recombinants. But if the distance between the genes will be less, then the recombination frequency will be less and you will have less number of recombinant. So since the recombinant is dependent upon the distance between the gene, you can use the number of recombinant to find out the distance between the genes. So what is the formula used? The formula used is the total number of recombinants divided by the total number of progeny multiplied with 100. So that will give you the distance between the genes. So here if you observe, there are 180 number of recombinants. So 180 divided by total number of progeny is how much? 820 plus 180, that is 1000 multiplied by 100. So the result will be 18. So the recombination frequency is 18% and the map distance is 18 map unit. So understood this, how the 2-point cross is used to calculate the map distance? So since we are considering only 2 genes here, we call it as a 2-point cross or 2-factor crossing. Similarly, we can have 3-factor crossing also, where 3 genes are utilized to find out the distance between the different genes that are located on the same chromosome. So here is another example. So here is another example that you can see. We have different phenotypic combinations here. Okay. So always remember, as I told you, the plus actually indicates a wild type gene. Okay. And the recessive character or the mutant character is expressed by the means whatever sign is given there. So here we are considering 3 characters. One is called echinus eyes, then cross venless wing and skewed bristles. So 3 characters are there which are located on the same chromosome. So the echinus eyes is represented by EC, the cross venless wing is represented by CB, and the skewed bristles are represented by SC. So SC, EC and CB all three of them are located on the same chromosome. So now, based on this data, you have to find out what is the distance, what is the order of the gene, and what is the distance between the respective genes. Okay. So here you are crossing a double mutant female with that of a wild type male. So since it is a wild type male, so it will have a normal eye, means it will not have echinus eye, it will have normal red color eye. Then it will have normal wings. Okay, CB, it will not have CB plus, it will not have cross venless wing, it will have normal wing, and it will also have the skewed, like the mutant is having skewed bristles, but this individual will have the normal bristles. Okay. So now, if you look carefully, so what kind of gametes you can expect from this? So this will give you a single type of gamete because both the chromosomes are having the same genotype. So you will have a SC, EC and CV combination from this. And here also it is a male. So this is actually representing a Y chromosome, but you will have this gamete. Okay. So the female in the F1 progeny will be a heterozygous because it will have the wild type character from the father and the mutant characters from the mother. So this heterozygous can actually form eight different type of gamete. So just let me show you a simpler form of this. Okay, this is visible to all. This one actually very important if you want to understand the 3-point cross. So always the 3-point cross is carried out between a hybrid and a double recessive mutant. So the double recessive mutant is nothing, but it will be having 2 recessive copies of each gene, small a, small b, small c. So you can have only one type of gamete from this. All the gamete that is produced from this double recessive mutant will carry the genotype small a, small b, small c. But this hybrid, which is having heterozygous genotype for all the 3 genes, can have 8 different type of gamete. So what could be the possible 8 different type of gamete? Now first gamete could be there is no crossing over. So what combinations will be there in the gamete? It will be A, B, C, capital A, B, C and small a, B, C. Okay. So like this. So this is called as the parental genotype because in parent also the same genotype combination was there. Now, what could be the other possible? Now, there could be a single crossover between the gene A and B. So if there will be a single crossover between gene A and B, the A will move to this chromosome and the capital A will come to this chromosome. So you will have 2 different gametic combination where it is a small a capital BC, capital A small bc. So this is a product of single crossover, that is SCO, single crossover product between the gene A and B. Similarly, there could be a single crossover between gene B and C. So if there is a crossing over between B and C, what kind of gamete you will get? Now there will be capital AB, small c, small ab, capital C. So this is again product of another crossover. Now the last part is it could be a crossover between 2 genes, means 2 crossovers might occur simultaneously where there is a crossing over between A and B also, there is a crossing over between B and C also. This is what is called as a double crossover product or DCO. Okay. So here what you can observe, these terminal parts will be exchanged. So the capital A and C will come to this chromosome and the small a and c from this chromosome will move to the other chromosome. So these are the 8 different type of gamete that you will get if there is a heterozygous trihybrid, heterozygous 3-point cross. So this part everybody understood or not? Because based on this only you will have this calculation and you have to understand what is the interference and coincidence in 3-point crossover. So if anyone did not understand, you can ask me. I will try to explain again. Okay, fine. Then let me assume that you have understood this. So let's come back to the question. So here, if you look into the data, there are two huge numbers mentioned here. Okay. So one is representing all the wild type phenotype, and the other one is representing the skewed, echinus, and cross manless. That means this is the recessive character. So both of these characters actually resemble the parental type. So this number, which is actually highest in any particular cross, is actually representing a parental combination. Okay. Similarly, you can observe that there are two numbers which are very low in number, two phenotypes which are very low in number. So because the number of progeny with this phenotype is only 1 and 1. Okay. So this is actually representing the double crossover or DCO, because in order to have a double crossover progeny, there should be two crossovers. So naturally, two crossovers are not very much allowed for a chromosome. So this kind of occurrence will be very low. The frequency of this kind of double crossover will be always the lowest. So these two progeny or these two classes are representing a double crossover product. Okay. Now, in between whatever rest of the combinations you are observing, they are actually the product of single crossover. Now, actually here we are not sure what is the arrangement of the gene. So first we have to find out exactly in what order the genes are arranged. Okay. So for that we have to analyze, or we have to compare the genotype of a parent and the genotype of a double crossover product. So let me do it for you. This is actually a little bit tricky thing. You have to be very careful, to be very focused in order to understand this part. So here one of the characters is what? Let me SC. Sorry. Actually, this genetics, it is very difficult to teach without paper and pen or chalk and board. Okay. So we have one combination, that is SC, then EC, then CV. Okay. And what is one of the double crossover genotype you are having? So it is echinus, means it is only having E and C. The rest of the characters are what? SC plus and CV plus. Okay. So whatever is not written means this is indicating that they are having the wild type character for the rest of the two phenotype. So since this middle gene is same and only these two genes have exchanged the part, the order of the gene is correct. Okay. Or let me take another little bit simpler example. This might be a little bit confusing to you. So let me show you a very simple example which I have cited here. Okay. This one, this is visible to all of you, this slide?
Yes, ma'am.
Okay. So here let's understand actually what does this sequence mean. Now look at the parental sequence. So parental sequence, so what is the parental genotype here? P plus, J plus, R plus. Okay. And can you tell me what is the double crossover class here? See, in any kind of problem you are having like this, always the highest number will represent the parental genotype, because when the gametogenesis is occur, even if the recombination does not occur, still the gametogenesis will take place. So most of the gametes will have the normal parental combination, even if there is no recombination. So always the highest number will represent the parental genotypic combination. So here the parental genotypic combination is what? P plus J plus R plus. And always, as I told you, the double crossover is very difficult to occur. So always the lowest number progeny class will represent the double crossover. So here what is the genotype of the double crossover? Double crossover is P, J plus R. Okay. Now, if you compare these two, okay, so compare one of the progeny, one of the parental genotype with that of a normal double crossover genotype. So the parental genotype is P plus J plus R plus, or what is the other parental genotype? That is P, J and R. So if you compare these two, tell me which two genes are actually remaining constant and which gene is actually different means like you saw here in the previous slide. This is actually a bit difficult to explain without writing much. Okay. So here if you observe carefully this slide, okay. So what is happening in case of double crossover? The gene that is present in the middle is where it is. Only the terminal genes are changing the position. Okay. So if you compare this gene, this combination ABC with a parental combination. So what you are observing? The middle gene is where it is. Compare these two. The two genes, that is A and C, that were capital in the parent has become now what? Small in comparison to the B gene. So the gene where it is, if it is like that, that is what is the gene which is present in the middle. So here the gene B is present in the middle. So by comparing with a double crossover genotype with a parental combination, you can find out actually which gene should be present in the middle. Okay, so that is how order of the gene is determined. So here in a parental combination, the capital A and C were present with the capital B. But in the double crossover, what you are observing? Small A and C are present with the capital B. So this means the capital B is present in the middle. Similarly, if we look into this case, so just compare the parent with that of the double crossover and tell me what you expect, which gene should be present in the middle.
J plus.
What about others?
As you said,
Everyone?
the terminals are changing and this middle one are always same.
Yes.
So here
See,
as I can
here, exactly. Here, since if you compare these two, the P and R where they were, but only which gene is changing the position? J. Instead of J, what you are having? J plus. So definitely J is present in the middle. So what I am trying to say
Ma'am,
is
there is
always,
4 type of genotype. So,
no,
which of
four,
them we
not
should
four. There are
consider?
8 different genotypes are there, not 4. You can see
Ma'am,
there are 8 different
parental
genotypes.
two, type 2, 2 and double crossover 2 type. So there
Yes.
is 4 type genotype. So we should consider 4 genotype to evaluate the arrangement.
No, yes. In order to find the order of the gene, you can compare any two of these. Means one from the double crossover, one from the parental combination. From there, you can easily find out which gene is present in the middle. So why I am telling you this? Because in all the problems, you will here actually the genes are present in the actual order. But in few cases, what you will observe, the genes will not be present in the order. They will be written randomly. First, you have to find out the order of the genes. After you find out the order of the genes, you have to arrange them on the chromosome. Then you can find out the single crossover between the respective genes, and then only you will be able to calculate the distance between the genes. Okay, so here fortunately we are having the genes, they are in the actual order. The same thing is happening here
so
also.
ma'am, one thing, like by the number also we can find out now which one is parental and which one is a double
Yes, by the
crossover
number
and
you can
combination.
find which one is the parental and which one is the double crossover. But you
Only
cannot
double or
find what is
single
the order of the
crossover.
gene.
A single crossover is like it's a bit hard to find, but double crossover we can find, right?
Yes, because that will be occurring in the lowest number. Okay. So
Okay.
from
Okay.
number
Thank
you can find which is the combination, but that does not give you, means that does not actually depict the order. So in order to find the order, you have to compare the sequence of the parent with the double crossover. Okay. Because as I saw you in this 14th slide, as you saw in the 14th slide. Okay. So here, sorry, 14th slide, not 14th. Okay. See, as I show you here in the 10th slide. Okay, so this is actually giving you the concept why you should be considering this with this one. Because until and unless you find the actual order of the gene, you cannot find the distance between the genes. Okay. So I have not cited a problem like that, but you will come across the problem like that where the genes will not be present in the order. So first you need to determine the order of the gene, then you can calculate the distance between the genes. So let's move to the calculation part. So here also for whatever problem is given in your topic, that in that case also the genes are present in actual order. So we don't have to worry much. So next what you have to do, you have to find what is the single crossover product. Okay. So here, so here, as you saw, these two are the means parental combination. These two are the double crossovers. Okay. So in between what you have, this is a skewed and this is echinus and cross-veinless. So skewed means this is only SC. The rest are EV plus, SC plus and EV plus. Okay. So these two actually giving rise the single crossover products in between. Or let me explain you the last one only, then you can easily find out or correlate with this one because the numbers are a bit difficult here. This one we can consider. So here also it is clearly mentioned which one is the single crossover product and which one is the double crossover product. So just let me tell you the calculation part. So here, if I want to calculate what is the distance between the genes. So now if I want to arrange the genes, so how the genes should be arranged? It is like P, J and R. So what is the single crossover product between P and J? Here, okay, because J and R as they are, but only the P has been exchanged here. So 46 plus 52 plus what is the the double crossover? 4 plus 2, that is 6. So just tell me how much it is, the calculation. 42 plus 52 plus 6. 104, I guess.
Madam, please explain how we are getting the numbers.
Which
How
number?
we are, these numbers 179, 173.
No,
How we
this
are
will be
getting this?
always given in your problem. So this is actually means, see, I gave you an example here, right? Wait. This is actually experimental result. Okay, so if you carry out an experiment where you have these kind of genetic combinations like you saw in this example I showed you. Here if you see, okay, so here they have carried out an test cross here. So from here only you are getting the numbers. So that is another example which I am citing because that is a little bit simpler. Understood now how you are getting the result? In the previous slide also you saw they were actually a cross was there, and that is how you got the number.
Yes.
Here, understood now from where you are
Yes.
getting the number. So like this, if you carry out the cross in a laboratory in an experiment, you will be getting the numbers like this, from where you need to analyze them. Okay, so let's go back to that problem. Let me explain that problem only. Later you can refer to this problem. So here the distance between the P and J, how much I calculated, that is 220.8. That part everyone understood? So since we are calculating the distance between P and J, okay, so we have to take the crossover between P and J, which is nothing but 46 and 52. Then the double crossover. So together we are getting 104 divided by the formula is total number of progeny, that is 500 into 100. So 104 by 5, that is equal to how much? 20.8. Is it clear to all, or anyone have any doubt up to this?
Ma'am, for you added 46, 52,
46
and
plus
then
52 plus double crossover, because when the double crossover is happening again, the P and J, they are also means the crossing over is also happening between them, so we have to add the double crossover in that also, because
okay.
every time the double crossover is happening, the single cross means it is also a recombination between P and J. So we have to include all the recombinants or all the recombination process that is happening between P and J. Am I clear now?
Yes, ma'am. Yes.
So why I am adding the double crossover? Okay. Similarly, in order to find the distance between J and R, all of you try to calculate what will be the distance between J and R.
Here P stands for parental, R stands for recombination,
No, no, no.
recombinant,
It is no, no,
and
no.
a
It
J
is the
stands for
name of
what?
the gene. See, this P,
Name
R,
of the
and J.
gene.
Yes, these are the name of the gene. Okay, this is a hypothetical example actually. Hypothetical means this gene does not exist. We are just taking them as an example. Okay. So just tell me what will be the distance between J and R using the same formula, same concept. So what are the crossover product between J and R?
10.
This one. 22 plus 22.
22 plus 22 plus 2.
No, plus 4, 6 you have to add.
Acha, 4 and 2 both we have to add.
Both you have to add, yes.
Okay.
10.
Yes, the distance will be 10 and it is represented by a unit called map unit or centimorgan. Small c, capital M. Actually, this is one of the means most important concept of the entire genetics, like how to determine the order
Ma'am,
of the gene.
how to find out the answer? Distance of distance of two gene. What question you asking before?
This is the
How we
formula you
calculate
are.
the formula? The formula clearly not so.
Okay, the formula is, okay, you write down, I am telling you the formula, you write down if you have notepad and like paper and pen. The total number of recombinant divided by the total number of progeny. Sorry, I couldn't hear you.
Yes, ma'am. The total number of recombinant
Divided by total number of progeny.
total number of progeny
Multiplied with 100 into 100.
into 100.
That is the formula to find out the distance between the genes. So, Sneha, if you don't have any problem, I would like to explain this in ODIA once again. So it will be easier for rest of the people to grab.
Yeah, ma'am. Sure, sure,
Okay,
sure.
okay, okay. सो अच्छा बाकी मन ओडिया रे कहे ताकि तुमको इजी हम बुझापी कि देख आमर जिते बड़े 3 पॉइंट क्रॉस माने कौन आम बुझुचे कि वी आर कंसीडरिंग 3 जीन साइमल्टेनियसली व्हिच आर लोकेटेड ऑन द सेम क्रोमोसोम ला मुझे पूरा एकदम बिगिनिंग रू बुझाऊँ जदि कहार किसी प्रॉब्लम हम पचार यहाँ गोटे बहुत ही इंपोर्टेंट कॉन्सेप्ट सो जिते भी टाइम लगू मुझे चाहूँगी कि बुझी जान समस्ते सो जमती एटा मो एग्जांपल नहीं
थैंक यू
ला देखो एटा जमती एग्जांपल नहीं 3 पॉइंट क्रॉस रे आमे कौन करूचे ना आमे गोटे ऑर्गेनिज्म नहींचे जो थरे 3टा जीन अच्छी कौन 3टा जीन अच्छी ए बी और सी ला सब बेले जेतेबेले हमर जीन डिस्टेंस आमे कैलकुलेट करे सेतेबेले आमे कौन करिबा ना फर्स्ट गोटे पैरेंटल कॉम्बिनेशन आमे नबा जो थरे दीटा को कौन हितबे होमोजाइगस डोमिनेंट और होमोजाइगस रिसेसिव ला माने कंट्रास्टिंग कैरेक्टर सो पूरा फर्स्ट पैरेंटल कैरेक्टर टा कौन थिबो कैपिटल ए कैपिटल बी कैपिटल सी कैपिटल ए कैपिटल बी कैपिटल सी सेमती तार माने जो अपोजिट पैरेंट टा कौन थिबो ना कंप्लीटली रिसेसिव हेतिबो कौन स्मॉल ए स्मॉल बी स्मॉल सी स्मॉल ए स्मॉल बी स्मॉल सी ला नाउ अच्छा ए जो जमती तुमको माने आमे काली जो पढ़ी मेंडेलिज्म जो टी टी रो कांसेप्ट माने तो जेतले गैमेटोजेनेसिस होतला हमको कोन गैमेट मिलुतला जो थरे टी टी अछि कोन गैमेट मिलिबो
टॉल
नहीं केवल कैपिटल टी कारण आउ किछि वेरिएशन ही नहीं हला सेमती जदी गोटे जीनोटाइप अछि स्मॉल टी तादरो कोन गैमेट तयारी हो केवल स्मॉल टी ही कारण आउ
स्मॉल
किछि
टी
वेरिएशन नहीं बट जदि गोटे जीनोटाइप अछि कैपिटल टी एंड स्मॉल टी ताल तुमको कौन टाइप रो गैमेट्स मिलबे ना क्रोमोसोम जेतले सेपरेट होउची 50% गैमेट रे कौन जाई पारे कैपिटल टी 50% गैमेट रे कौन जाई पारे स्मॉल टी ए पार्ट टा बुझी करछ ना नहीं समस्ती एटा फंडामेंटल जिंस किंतु एटा न जानले आगो को किछि बुझेबोनी हला तो एटा जाय क्लियर समस्ते एवरीवन क्लियर
यस मैम
जेमती एटा सिना गोटे जीन अछि बोले हमें एमती इजीली जानि दोछे जेते बेले देयर आर मोर देन वन जीन जेमती हमें डाई हाइब्रिड रे देखितले कोन थिला वाई वाई आर आर जो थिला सो सेमती एथरो कोन कॉम्बिनेशन हबो खाली वाई खाली आर सेमती स्मॉल वाई स्मॉल आर किंतु जदि डाई हाइब्रिड माने जदि हेटेरोजाइगस थिबो ताले कोन वेरियस कॉम्बिनेशन सेही परिबो ना नहीं चारि प्रकार गैमेट हेई परिबे कोन कैपिटल वाई कैपिटल आर कैपिटल वाई स्मॉल वाई कैपिटल आर स्मॉल आर so these are the different type of gametic combinations which are possible सेमती जेतले हमें ट्राई हाइब्रिड माने जो तीनटी क्या देखुचे बा तीन 3 पॉइंट क्रॉस देखुचे जदि फर्स्ट पेरेंट रे एमती अछि कॉम्बिनेशन कैपिटल एबीसी एडा भी अछि कैपिटल एबीसी तार ऑपोजिट पेरेंट अछि डबल रिसेसिव पेरेंट सबले हमर निया हुए कारण हमर इजीली हमें हेटेरोजाइगस पेरेंट रे रिकॉम्बिनेशन को ट्रेस करि परिबा बोली सो ए पेरेंट रो कोन प्रकार गैमेट तुमको मिलिबो ना जदि भी आंकर रिकॉम्बिनेशन हुए किछि नुवा कॉम्बिनेशन अछि कि नहीं कारण दीटा कर एलीलिक कॉन्स्टिट्यूएंट सेम अछि तो तुम कौन मिलला गैमेटिक कॉन्स्टिट्यूएंट कैपिटल ए कैपिटल बी कैपिटल सी हला सेमती ए पेरेंट रो कौन गैमेट्स तयारी हेई परिबो को वेराइटी रो ना खाली स्मॉल ए बी सी स्मॉल ए स्मॉल बी स्मॉल सी नाउ जेतले ए दीटा को गैमेट फ्यूजन हैला सपोज दिस इज द स्पर्मेटोजोआ दिस इज द ओवम ओके सो तमरो पेरेंट में हेटेरोजाइगस जो प्रोजेनी होबा तार कोन होबा जीनोटाइप किंतु कैपिटल ए कैपिटल बी स्मॉल बी कैपिटल सी स्मॉल सी सो अप टू दिस समझते क्लियर की नहीं
यस मैम
तो देखो एग्जैक्टली से जिनसटा एठी लिखाई छी कि नहीं कैपिटल ए कैपिटल बी कैपिटल सी स्मॉल सी अच्छा एडी आउ गोटे जिनस नोट करो कौन ना एबीसी जो अच्छे सबुतक कैपिटल एबीसी गोटे क्रोमोसोम रे अच्छे स्मॉल एबीसी गोटे क्रोमोसोम रे अच्छे सो ए जिनसटा का हमर कोन कह जाय ना सीस फेज बा कपलिंग फेज जो थरे ऑल द डोमिनेंट कैरेक्टर्स आर प्रेजेंट ऑन वन क्रोमोसोम all the recessive characters are present on the other chromosome okay but bele bele kon he pare na eithre a capital A thibo स्मॉल बी थिबो कैपिटल सी थिबो सो से जगह आमे ताको ट्रांस अरेंजमेंट बोली कहबा जोथरे कि स्मॉल और कैपिटल मिसी की थाई पारे बट एनीवेज हमर एठी सेमति किछि नाही जेमति एई जगह रे देखुछ दिस इज ए ट्रांस अरेंजमेंट काहेकि ना सब डोमिनेंट और रिसेसिव गुडा एका संगे नाहंती दे आर मिक्स एंड मैच्ड ହେଲା सो जो जगह रे डोमिनेंट गोटे क्रोमोसोम रे रिसेसिव गोटे क्रोमोसोम रे थिबे दैट इज व्हाट इज कॉल्ड एज ए सीस अरेंजमेंट सो ए पेरेंट रे जितले गैमेटोजेनेसिस हबो ताहले कोन पॉसिबिलिटी अछि ना मे बी दीटा को क्रोमोसोम रे रिकॉम्बिनेशन बिल्कुल हबो ही नाही सो रिकॉम्बिनेशन न हेले कोन हबो ना जेमती अछि माने कॉम्बिनेशन आलेले कॉम्बिनेशन सेमती ही सेपरेट हे जिवे सो कोन हैला ना गोटे गैमेट को पळैला कैपिटल एबीसी तार ऑपोजिट को कोन जिवो स्मॉल एबीसी किंबा पॉसिबिलिटी अछि कोन ना गोटे ए और ए भितरे रिकॉम्बिनेशन हबो सो से जगह कोन ना सिंगल क्रॉसओवर हेछि सो कैपिटल ए स्मॉल बी सी संग पळे आछि स्मॉल ए कैपिटल बी सी पाखो पळे जेछि सिमिलरली आउ गोटे कोन पॉसिबिलिटी अछि ना केवल रिकॉम्बिनेशन अछि बी और सी भितरे सो एमती टाइप रो कॉम्बिनेशन तयारी हगला कैपिटल ए बी सहित स्मॉल ए बी सहित कैपिटल सी किंबा आउ गोटे पॉसिबिलिटी कोन अछि ना जोथरे दी थरो क्रॉसओवर हेछि ए और बी भितर भी क्रॉसिंग ओवर हेछि प्लस बी एंड सी भितर भी क्रॉसओवर हेछि तो सेही जगह रे कोन तमरे गेला डबल क्रॉसओवर अच्छा एबे देखो जदि हमें रिकॉम्बिनेशन बाहर करिवा बा डिस्टेंस बाहर करिवा को चाहेबा बिटवीन ए एंड बी सो ए एंड बी भितर रो रिकॉम्बिनेंट की को जगह रे ए और बी भितर रिकॉम्बिनेशन हेछि एई जगह रे ओके अच्छा जितले एठी भी ए एंड बी भितर रिकॉम्बिनेशन हेछि कि नाही डबल क्रॉसओवर
यस मैम
हेछि सेथि पई आमे जेतले टोटल नंबर ऑफ रिकॉम्बिनेंट ऐड करिबा फॉर डिस्टेंस बिटवीन ए एंड बी वी हैव टू ऐड दिस प्लस दिस एबे बुझिलो कि हमें डबल क्रॉसओवर टा को काहे के ऐड करूचे because when the double crossover is happening a crossover is also happening between both the genes so that is why this is also a recombination product between the gene A and B सेमती यार भीतर जितल डिस्टेंस बाहर करिबा recombination product between these two plus the double recombinant so together they will represent the recombinant between B and C am I clear up to this कि आउ कहार किछि डाउट्स अछि एते बजाय
Yes, ma'am.
Okay. So एबे आसीबा से कैलकुलेशन पार्ट को सो हमें एठी कोन कैलकुलेशन करथिले द डिस्टेंस बिटवीन P एंड J केते बाहर करथिले 20.8 और डिस्टेंस बिटवीन J एंड R केते कैलकुलेट करथिले 10 सो एबे जदि म एटा मैप रे शो करबी केमति लिखिबी this is P this is J this is R ला so तमे जतले मैप टा कंस्ट्रक्ट करिवो ना एटा 10 अछि एटा 20 अछि so distance between P and J should be twice that of the distance between J and R ला so सेमति प्रकार ड्रा करिवो 20.8 so up to this clear केमती मैप डिस्टेंस कैलकुलेट करिवो एबे फर्दर जोठी भी आसीबो क्वेश्चन यू कैन कैलकुलेट आई होप
Yes, ma'am.
एठी आउ गोटे हमें कांसेप्ट जिबा कोन ना कोइंसिडेंस ओके कोएफिशिएंट ऑफ कोइंसिडेंस और COC एंड इंटरफेरेंस सो एटा कोन बुझिबा अच्छा एटा तमे जो एक्सपेरिमेंटल गोटे एक्सपेरिमेंट करके रिजल्ट पाइछ ला सो ए जो जिनसटा देखो एटा ऑब्जर्व्ड ना एटा एक्सपेक्टेड मते एति कहला whether the double recombinant जोटा तमे देखो it is a observed result or this is an expected result कौन भाबुछ
Expected.
मैडम
एक्सपेक्टेड
ऑब्जर्व
अच्छा जे कहू ऑब्जर्व्ड काहे के ऑब्जर्व्ड जे कहू एक्सपेक्टेड काहे के एक्सपेक्टेड
It is observed because after experiment these numbers are comes.
Exactly. This is actually observed number of double crossover. La. But एबे हमें find out करिबा what could be the expected. Expected means theoretically what should have been the number of double crossover, but what you are actually observing. So the ratio between the observed number of double crossover and expected number of double crossover is known as coefficient of coincidence. So एबे आमे देखिबा आमे expected टा केमती पाइबा, expected value टा कोन होबा कथा हमरो ना एठी देखो आमे जो distance calculate करिचे. So the map distance is also another way of expressing the frequency of recombination. So जो 20.8 लिखिछो, this is the percentage or 20% is the frequency of recombination between P and J. Similarly, the frequency of recombination between J and R is 10%. That means out of 100 progeny, 20 progeny should be recombination product between P and J. Out of 100 progeny, 10 progeny should be the recombination product between J and R. But haba kotha, but actually kon hochi ba kete difference hochi, let's calculate that. अच्छा, so एबे हमर expected number of double crossover हमें केमती calculate करिबा ना, the distance between the genes, that is 20.8 into 10 divided by 100 into 100 multiplied by 500. So ये value टा calculate करलो कितने होची 20.8 into 10 into 500 divided by 100 into 100. Mobile
10.4.
10.4. Okay.
10.4.
10.4. Okay, fine. So actually 10.4 is actually our expected number of double crossover. So how many double crossover you should expect? 10.4, let's say it should be 10. But actually how many double crossover you are observing experimentally? 4 plus 2, how much?
6.
6. So this ratio is actually representing the coefficient of coincidence. What is the coefficient of coincidence? The expected number of double crossover, observed number of double crossover divided by expected number of double crossover. Am I clear up to this? A part ta ke bujhi parlo na bujhi parlo ni?
Ma'am, how to
अच्छा, तो अब थोड़ा एक्सप्लेन कर दूची. देखो थियोरेटिकली हमें कौन देखुचे डिस्टेंस बिटवीन P एंड J कौन अछि 20.8 अछि ना मैप यूनिट
आज्ञा मैम
आउ डिस्टेंस बिटवीन J एंड R कौन अछि 10
10
मैप
अछि.
यूनिट अछि. ओके,
okay.
सो तमरो केते फ्रीक्वेंटली P एंड J रिकम्बाइन करिया कथा ना 20.8 माने अप्रोक्सीमेटली 21 धरिनियो. So 21 times तमरो बा 21 should be the percentage of recombination between P and J. So 20.8. And what should be the recombination frequency between J and R? 10 times. So you are multiplying both of them. अच्छा, गोटे प्रोजेनी रे तमे P and J भितरे केते परसेंटेज रिकॉम्बिनेशन एक्सपेक्ट करछो 21%. आउ J और R भितरे केते रिकॉम्बिनेशन एक्सपेक्ट करछो 10%. सो गोटे प्रोजेनी रे P और R भितरे केते रिकॉम्बिनेशन होबा कथा बा डबल रिकॉम्बिनेशन केते टाइम होबा कथा ना P and J, P और J भितरे into J and R भितरे. सो 20.8 into 10. जेहेतु हमें एटा परसेंटेज रे नोचे वी आर डिवाइडिंग इट बाय 100. अच्छा, टोटल नंबर ऑफ प्रोजेनी तमरो केते अछि एठी एक्सपेरिमेंट केते जण प्रोजेनी तमे पाइछो टोटल 500. तो जदि गोटे
500
प्रोजेनी रे दिस शुड बी द फ्रीक्वेंसी ऑफ रिकॉम्बिनेशन 500 प्रोजेनी रे केते होबा कथा सिंपली मल्टीप्लाई कर दियो 500 रे हला ताले केते मिलला रिजल्ट 10.4. सो थियोरेटिकली तुमको 10.4 टा डबल रिकॉम्बिनेशन मिलवा कथा बट एक्चुअली तुम केते पाउछो मात्र 6 टा ना. सो से जो डिफरेंस बा से जो रेशियोटा मिला 6 डिवाइडेड बाय 10.4 दैट इज एक्चुअली कॉल्ड एज कोएफिशिएंट ऑफ कोइंसिडेंस एबे बुझ पारलो ना पुणी आउ कार डाउट्स रहिला ए पार्ट बुझ एक्चुअली एटा डिफिकल्ट अछि सो पचलले किछि प्रॉब्लम नै सो नाउ अंडरस्टूड हाउ वी आर कैलकुलेटिंग द एक्सपेक्टेड नंबर एक्सपेक्टेड होची हमें थियोरेटिकली केति की एक्सपेक्ट करछे जेहेतु एटा 20 पॉइंट माने 21 मैप यूनिट डिस्टेंस रे अछि 21 टाइम्स या भीतर रिकॉम्बिनेशन होबा कथा सेमति ये जेहेतु माने 10 मैप यूनिट डिस्टेंस रे अछि 10 टाइम्स या भीतर रिकॉम्बिनेशन होबा कथा आ टोटल डबल रिकॉम्बिनेशन केते होबा कथा ना यार and तार since and आमे यूज करछे so we are using multiplication ହେଲା सो आउ केते रे गोटे रे जदी आमे एति की एक्सपेक्ट करछे 20.8 by 100 10 by 100 ताले 500 रे केते हबा कथा ना सिंपली मल्टीप्लाई इट विथ 500 सो 500 हम कोठ आनजे एटा नंबर ऑफ प्रोजेनी रु आनजे हैला जदी एट नंबर ऑफ प्रोजेनी 1000 थिबो ताले यू हैव टू मल्टीप्लाई इट विथ 1000 सो दिस इज सिंपली रिप्रेजेंटिंग द टोटल नंबर ऑफ प्रोजेनी so जो आमे पाइलो केते 10.4 so 6 divided by 10.4 that is 0.57 is actually your coefficient of coincidence so everybody understood up to this or not
yes ma'am
na kaha doubts achi okay now we have another term called as interference so interference is kona now 1 minus coefficient of coincidence okay so 1 minus amar eti coefficient of coincidence kete bahla na 6 by 10.4 jodi tame karibo the result will be I think 0.57 something will be the result la tale interference kete hobo ना 1 minus 0.57 हेले केते हबो एटा 43 हबो बोधे
0.43
or in other terms we can call it केते 43% अच्छा इंटरफेरेंस रा माने कोन हम कहि दोची इंटरफेरेंस रा माने होची always the second crossover is inhibited by the first crossover or जेतले हमें डबल क्रॉसओवर देखछे कोन होची हमर दीटा क्रॉसओवर होची ना फर्स्ट ए जगह रे क्रॉसओवर हैला पुनी ता नियर बाय रीजन रे क्रॉसओवर हैला सो इंटरफेरेंस रा माने होची कि केते परसेंटेज रे द फर्स्ट क्रॉसओवर इज ट्राइंग टू इनहिबिट द ऑकरेंस ऑफ द सेकंड क्रॉसओवर सो ए जगह रे तमरो इंटरफेरेंस केते अछि 43% अछि जेतले तमरो इंटरफेरेंस जदी 0 if interference is 0 then what is coefficient of coincidence kete hobo कहलो जदी interference ता 0 हे जिवो coefficient of coincidence ता केते होबो 1 okay तार माने कोन ना observed number of double crossover will be equal to कोन expected number of double crossover is not it because coefficient of coincidence हमें कौन लिखुचे observed number of double crossover divided by expected number of double crossover so when the interference will be 0 then your observed number of double crossover will be equal to expected number of double crossover similarly if interference will be 100% ताहले कौन होबो तमरो outcome ना coefficient of coincidence केते हिजबो 0 माने डबल क्रॉसओवर विल बी 0 एबे बुझ पालो इंटरफेरेंस रो कोन इंपोर्टेंस सो इंटरफेरेंस इज एक्चुअली कंट्रोलिंग what should be at or at what frequency the double crossover should be produced so more is the interference the less will be the observed number of double crossover am I clear up to this कि आउ कहार कोन डाउट्स रहिला
मैम आउ थरे से इंटरफेरेंस टा टे बुझे दियंतु ना
अच्छा देखो इंटरफेरेंस रा माने होची एक्चुअली अच्छा डबल क्रॉसओवर माने बुझछ जो एक्सप्लेन कली कि साइमल्टेनियसली तीनटा जीन भितरे दी थर क्रॉसओवर होची तो इंटरफेरेंस रा टेंडेंसी कोन ना इट विल ऑलवेज ट्राई टू इनहिबिट द सेकंड क्रॉसओवर माने जदी क्रोमोसोमल इंटरफेरेंस जदी बेसी हबो ताहले ये सेकंड क्रॉसओवर होबा को दबनी सो हायर इज द डिग्री ऑफ इंटरफेरेंस less will be the number of double crossover. Mane they are inversely related. Hela? So more will be the number of higher will be the interference level, less will be the number of double crossover. So এই জায়গায় আমি যেমতি দেখিলে কোন না 43% হচ্ছে ইন্টারফিয়ারেন্স সো সেইথিপাই কি তোমার অবজর্ভ নাম্বার অফ ডাবল ক্রসওভার ইজ অ্যাপ্রক্সিমেটলি মানে অ্যাপ্রক্সিমেটলি ক্লোজ টু দ্য এক্সপেক্টেড নাম্বার অফ ডাবল ক্রসওভার 6 ଆଉ 10 সো বহুত বেশি ডিফরেন্স নাই না बट जो जगह इंटरफेरेंस जदि बहुत बेसी हम जमती आम प्रीवियस एग्जांपलटा जो देखा ये अच्छी जोटा एक्चुअली तुम बुक रो जोटा एक्सपेरिमेंटा निया हिची एटी देखो ये जो आमे प्रॉब्लमटा नेइथिले एटी देखो डिस्टेंस आमे केते से कैलकुलेट करछि केते डिस्टेंस बिटवीन द फर्स्ट टू जीन्स इज 9.1 सेंटीमॉर्गन सेकंड जीन रो डिस्टेंस केते कैलकुलेट करछि 10.5 सेंटीमॉर्गन ताहले ए जगह हमरो डबल क्रॉसओवर हम एक्सपेक्ट केते करबा कथा ना डबल क्रॉसओवर एक्सपेक्टेशन शुड बी एडी कैलकुलेट करके देछि तमरो से कोडे वाला हां एडी सॉरी देखो नंबर ऑफ डबल क्रॉसओवर तुमरो केते से कैलकुलेट करके देछि ना 0.0 सॉरी ए डेटा को मल्टीप्लाई कर देले तुम पाइबो नंबर ऑफ डबल क्रॉसओवर हैला सो एटा तुमरो जो ऑब्जर्व नंबर ऑफ डबल क्रॉसओवर सॉरी एक्सपेक्टेड नंबर ऑफ डबल क्रॉसओवर केते देछि ना 0.0095 मल्टीप्लाइड बाय 0.0006 जो डेटा को अछि तुमरो नंबर एटा कैलकुलेटेड विथ मल्टीप्लाइड विथ टोटल नंबर ऑफ प्रोजेनी जदी करिवो तमे सो दैट विल गिव यू द एक्सपेक्टेड नंबर ऑफ डबल क्रॉसओवर हैला बट तमे ए जगह रे देखिबो केते तुमरो ऑब्जर्व नंबर ऑफ डबल क्रॉसओवर इज ओनली 2 सो एठी तमे कैलकुलेट करके देखि पारिबो इंटरफेरेंस से केते कैलकुलेट करछि 93.7%. सो तमर एक्सपेक्टेड एक्चुअली एठी हबा कथा डबल क्रॉसओवर 30 बट तमे ऑब्जर्व केते करछो ओनली 2 मान तमर हबा कथा केतेटा डबल क्रॉसओवर 30 टा पाखापाखि डबल क्रॉसओवर प्रोजेनी हबा कथा किंतु तमे एक्चुअली केतेटा देखुछो ओनली दीटा काहे कि ना एठी इंटरफेरेंस अछि 93% माने ऑलमोस्ट पूरा इंटरफेरेंस एते अच्छी से चाहू ही नहीं कि तुम डबल क्रॉसओवर हो एबे बुझ पालो इंटरफेरेंस रो माने कोन द इंटरफेरेंस विल ट्राई टू लिमिट द possibility of a double crossover so the less is the number of interference or the less is the degree of interference the higher will be the double crossover or higher will be the coefficient of coincidence or in other terms you can say the observed number of double crossover value will be close to the expected number of double crossover value. Okay, is it clear to all or आ गोटे दुई थर माने तमे एटा रिवाइज करो तापरे धीरे धीरे बुझ परिबो मे बी पूरा सडनली एटा मे बी टिके डिफिकल्ट होतबा कैच करिया पई किंतु गोटे दुई थर तमे निजे रीडिंग करबो रिवाइज करबो तो तापरे कोरिलेट करि परिबो यदि एक्सप्लेनेशन को टिके माने ये करिया पई चाहिबो
यस मैम
नले माने ये करो काली आउथरे मो आई कैन एक्सप्लेन टुमारो अगेन सो देखो एडी जेमति से कैलकुलेट करछि तांगर माने से जो सेम सॉरी जोटा हमें कैलकुलेट कले जमते हमें निजे कैलकुलेट करथले एटी सी भी बाहर करछि तारो कोएफिशिएंट ऑफ कोइंसिडेंस केते बाहर करछि 0.063 सो से हिसाब रे इंटरफेरेंस केते अछि बहुत ओके सो जेहेतु इंटरफेरेंस इज मोर द एक्सपेक्टेड नंबर ऑफ डबल क्रॉसओवर 30 थिला को तुम ऑब्जर्व करछो ओनली 2 जो जगह इंटरफेरेंस जदी कम थिबा एक्सपेक्टेड वैल्यू ऑब्जर्व वैल्यू विल बी क्लोज टू ईच अदर or the number of observed double crossover will be close to expected number of double crossover. Okay, then coming to the last part of this section, so that is your genetic mapping using the pedigree analysis. Okay, so it is a simple way I will try to explain it. Acha pedigree actually tomorrow gote अच्छी separate topic अच्छी pedigree रो बट इटा जस्ट जानी था पेडिग्री होचे हमर कोन जेतला हमें गोटे फैमिली केनो कोनोसी कैरेक्टर को फैमिली गोटे चार्ट रे हमें एक्सप्लेन करछे ना सो ए जगह रे देखिबो जदी तमे हमें कोन कोरिलेट करछे ना दीटा कैरेक्टर अछि गोटे होची नेल पैटेला सिंड्रोम आ गोटे होची ब्लड ग्रुप टाइप बी ओके
मैम can't understand please
Oh, sorry. I forgot. Okay, see, actually, here, in case of like as we saw in case of previous examples, we are calculating the map distance, etc., by carrying out various crosses. But that will not be applicable to higher animals. Like we cannot keep on crossing them, breeding them like this. So we have to adopt a different method or different means to predict the linkage or to predict the map distance between the genes in case of higher animals, like for example in humans. So what we are taking here, now we are considering the pedigree chart. Okay, so in pedigree chart, just means be a little bit attentive here. So what you can observe is the individual who is having a blood group B will have a nail patella syndrome. So nail patella syndrome means it will have a defective nail and some problem in the knee bone like that. Okay. So what we are observing is the B blood group is associated with the nail patella syndrome. Okay. Now, if you analyze the three generations of a particular family, what you can observe is sometimes there will be exceptions. So exceptions like you can observe this individual and this individual and this individual. The individual number 3, individual number 6 and individual number 12. So what exception you are observing? Now in the first case, it is having a blood group B, but does it have nail patella syndrome? It does not have, because the nail patella syndrome is represented by the colored boxes. Okay, so this is actually a recombinant, because the blood B should be associated with nail patella syndrome or B should be linked with the nail patella syndrome. But here you can observe the blood group B, although it is having, but it individual does not show nail patella syndrome. So means this is regarded as a recombinant. Similarly here, the individual 6, it is having a blood group, it does not have a blood group B, but still it is showing a nail patella syndrome. So again, this is also another representation of a recombinant. Similarly here, the last case, it is also having a blood group B, but it does not have the nail patella syndrome. Similarly, in the next generation, this is also having a blood group B, but it does not have the nail patella syndrome. So these 4 individuals are regarded as recombinants, because in their parents, the blood group B was associated with nail patella syndrome, but in the subsequent generation, in the subsequent progenies, there is no connection between them, or the two characters, they have been separated by recombination. So here, how we will calculate the map distance or the distance between the gene that controls the nail patella syndrome and the blood group? Now simply the total number of recombinant divided by total number of progeny, that is 4 divided by 13, which will be approximately 31%. Okay. So the distance between the two loci was found to be 31 centimorgan. So this is nothing but a simple condition where instead of using repeated crosses, we are using a pedigree chart to depict the relationship or the distance between the genes. Okay. So is there any question from any of you till now, or else we will move into a new chapter actually. So if there is any query from anyone from the previous chapter, now you can clarify, or else we will move to the next topic.
Ma'am, to revise it, we can follow the book provided from the university, right?
Sorry, I couldn't hear you properly.
Ma'am, I'm asking to revise it, all these things to revise. We can follow our download book that we have got.
I'm really sorry, I couldn't, it's not very clear actually. Could you please repeat once?
Okay. Am I audible, ma'am?
Yes, you are audible, but I couldn't understand your question actually.
Okay, I'm asking to revise all these things. We can follow our book that we have got.
Yes, your material is actually sufficient, but if you need some additional, more clarification, if you want, you can simply refer to Principles of Genetics by Gardner. It's like everything is illustrated in a very simplified manner there. Or you can also refer to a book. There is a book called I Genetics by Peter J. Russell. You can refer to that also.
Okay.
So both of the books are very simple, very illustrative, means clarity you will get. The concept means all the concepts will be explained very clearly.
Ma'am, the book's name again, please.
Okay, sorry, I couldn't hear. Can you speak one at a
The
time,
name
please?
of the books. Please repeat the name of the book.
Okay. One book is Principles of Genetics by Gardner. And
Another one.
another is Peter J. Russell, I Genetics. I, small I, Genetics by Peter J. Russell. But in my opinion, your material is actually more than sufficient. It includes summary from all these important books. Okay, so if you thoroughly go through this also, then that is also sufficient for you.
Okay, thank you, ma'am.
So, is there
Okay,
any
ma'am.
other question from anyone, or we will move to the next
No, ma'am.
concept?
Please proceed.
So next block actually we have mutation. Okay. So there is a very simple concept, mutation. So mutations means any change in the DNA sequence is regarded as a mutation. So we know DNA is made up of 4 alphabets, A, T, C, G, and all these alphabet they are arranged to give a particular sequence. The sequences can be depicted into 3-letter codons. The codons will be transcribed into mRNA codon, and which is actually translated into an amino acid that will form a polypeptide chain, or ultimately leading to the formation of a protein. The protein in turn will control or will be actually giving a particular characteristic or trait which we are calling as a phenotype. So any change in the sequence of DNA can affect the end product, that is the phenotype. So this process is called as your mutation. So as you see here, there is a normal DNA sequence or a normal gene is there. So you have a normal protein and you have the normal phenotype. For example, let's say the hemoglobin. Okay. So there is a HBB gene that encodes the beta globin chain, and you are having the normal shape RBCs. Now, if there is any change in the HBB gene, then ultimately you will have a defective, there will be change in the codon, and you will have a abnormal or a defective protein leading to altered phenotype of the RBC, that is the sickle cell RBC, and the phenotype developed will be sickle cell anemia. Now, one thing I would like to point out here, there is a difference between, okay, frequently you will come across a term called SNP, single nucleotide polymorphism. Okay. So here also you can observe that the DNA sequence of two individuals will be different, but that is not mutation. So it's not like every change in the DNA sequence will be regarded as a mutation, because in a population there could be variation of certain bases, variation of certain DNA bases among the individuals can exist. But so how you will differentiate a polymorphism or single nucleotide polymorphism from a mutation? Now based on the frequency. The frequency of a mutant gene will be always less than 1% within a population. Sorry, not 1%, it is now regarded as 6%, I guess. Okay. So less than 6% it will be. And also, the mutation is always associated with a abnormal phenotype. But for a single nucleotide polymorphism, there will be variation in the bases, but the bases will be present, the variations will be present in like 90% of the population. And also, due to the variation of the DNA base, no abnormality or no disease phenotype will be present in the population. So that is how you differentiate between a polymorphism and a mutation. Okay. So in simple terms, we can say the mutation should be less in number, Means the mutation frequency will be very low in a population, and also always the mutation is associated with a abnormal or with a disease phenotype or with a altered phenotype. But single nucleotide polymorphism, the base sequence might be different among the individuals, but the difference will occur in almost 99% of the population, and also the base change or the nucleotide change is not associated with any kind of abnormality. So this is a simple thing, basic thing which you should be knowing. Now mutations can be broadly divided into two different categories: point mutation and your chromosomal mutation. So point mutation actually refers to a mutation that occurs within the sequence of DNA or that affects usually a single gene. But chromosomal mutation means it will be affecting the chromosome number, okay, number as well as your structure. The chromosome number and structure both are affected, as well as since the chromosome can have multiple genes, so simultaneously many genes will be affected in a chromosomal mutation. So let's have a look at the single gene mutations. So there could be various types of single gene mutations. So you should remember the names as well and what does they mean. So there could be first as you can see, the first two type of mutations are called as base shift mutation. Okay. So in this case, what happens? Now there is a change in the base pair. Okay, like base substitution mutation. These are called where a purine base. Okay, so first let me tell you there are four type of bases in DNA. That is A, T, C, and G. So A and G, they represent the purine group of DNA base. C and T represent the pyrimidine group of DNA base. Okay, so A and G, when there is any replacement of the bases, like A is replaced by C, or A is replaced by T, G replaced by C, or G is replaced by T. So these kind of mutations are called as base substitution mutation. So base substitution could be two types. One is transition, other one is transversion. So when the substitution occurs within the same group, like A replacing B, G or G replacing A, or purine replacing purine, pyrimidine replacing pyrimidine, that is what is called as a transition mutation. If the mutation occurs between the two different groups, like a purine replacing pyrimidine and vice versa, that is what is called as a transversion mutation. Okay. Then based on how they will be affecting the amino acid sequence, four different type of mutations are there. One is called missense mutation, then nonsense mutation, then we have silent mutation, and we have neutral mutation. So missense, miss means always something wrong. So if due to change in the base or due to a change in the base pair, if a genetic codon is wrongly read, so it will definitely result in the formation of a wrong wrong amino acid. So when the mutation leads to formation of a wrong amino acid, that type of mutation is called a missense mutation. Means here the sense codon is wrong. Means there is a mistake in the sense codon. Okay. So like as you can see earlier, the sequence was AAA. Now the A has been replaced by G. So here earlier when the amino acid coded by A was lysine, now the amino acid coded is glutamine or glutamic acid. So because of this, there is a change in the amino acid. This kind of mutation is called as missense mutation. Similarly, nonsense mutation is when a sense codon is converted into a stop codon. So here AAA is converted into a TAA. So T will be U in case of mRNA. So it will be UAA. So UAA, UAG and UGA, all three are the stop codon. So here where there are more amino acids are to be synthesized, because of the conversion of a sense codon into the stop codon, the amino acid synthesis will stop in between and you will have an incomplete polypeptide chain. So this kind of mutation is called as a nonsense mutation. Okay. So next we have silent mutation and neutral mutation. So for neutral mutation, as the name indicates, the mutation will be there, but since the new amino acid synthesized is actually of similar character or similar property with that of the old amino acid, the protein structure will not be that much affected. Like the lysine and arginine both are basic positively charged amino acid. So if instead of lysine arginine is synthesized, the polypeptide chain will be functioning normally. So since the effect is not that much here, the mutation is called as neutral mutation. Now, sometimes due to degeneracy of the genetic code, that means multiple codons present for a single amino acid. Although there is a change at the DNA level, the same change cannot be observed at the protein level. Like the AAA also codes for lysine, AAG also codes for lysine. So if you look here, it can be seen that this is a mutation, but at the protein level there will be no observation because both the codons will be coding for the same amino acid. So since here mutation is silent, it is called as a silent mutation. Now, sometimes what might happen, there could be insertion and deletions. Like maybe by mistake, extra base is added, or sometimes when the when the replication of the DNA is taking place, a base might be removed, or means the DNA polymerase may by mistake leave one base. So in that case, it will be either addition or deletion, or we can say it will be insertion or deletion. So, in that case, the entire amino acid sequence will change following the point of mutation. Is there any question from anyone? You can ask.
No, ma'am.
No, Gloria, you have any question?
Sorry, ma'am.
Okay, no problem. So here, following the point of mutation, the entire amino acid sequence will change. So actually, the whatever codons are there in a sequence that is actually read by the tRNA, this is called as a open reading frame or a reading frame. So since following the point of mutation, the entire reading frame is changed. This is what is called as a frame shift mutation. Shift means change. So since the entire reading frame is changed following the point of mutation, this kind of mutation is called as a frame shift mutation. Okay. So I hope all the mutations types are clear. First, you have a base substitution where a purine can be replaced by purine or pyrimidine can be replaced by pyrimidine, or it could be vice versa. A purine can be replaced by pyrimidine also, pyrimidine can be replaced by purine. So based on this, you have either transition or transversion mutation, or based on how the codon is affected due to mutation, you can have either a missense mutation where a different amino acid will be synthesized, or you can have a nonsense mutation where a sense codon is converted into a stop codon and the protein synthesis will stop in between, or it could be a neutral mutation where due to mutation a similar property amino acid will be synthesized like lysine, arginine, or like aspartic acid and we can say glutamic acid, like that. So similar type of amino acid will be synthesized, so that will be a neutral mutation. Or it could be like due to degeneration of the genetic code, sometimes even if the codon will change, the amino acid will remain the same. So in that case the mutation remains silent, leading to a silent mutation. Or sometimes what can happen, a extra base might be added or an existing base might be deleted, that will change the entire reading frame, which is regarded as a frame shift mutation. So now just tell me which type of mutation you think is the most dangerous type from all the 6 types we discussed,
Frame
where you
shift
can
mutation.
observe the maximum effect of the mutation.
Maybe frame shift.
Frame shift, yes, because following the point of mutation, the entire sequence is changing, and means unfortunately, if this mutation occurs in like some earlier position of the gene, like for example, if let's say there are 145 polypeptide, 145 amino acid in a polypeptide. So if the change will occur at the second or third position, then following the point of mutation, the entire sequence will change. So that is actually the most disastrous type of mutation. Now coming to the chromosomal mutation, so there are actually 4 broad categories of chromosomal mutation. deletion. So simple as the name suggests, a part of the chromosome will be deleted. So one example of deletion mutation you can say, in our fifth chromosome in case of human beings, the short arm of the fifth chromosome is deleted, and because of which you have the child or the means individual will develop a like mewing of a cat kind of sound. I mean actually the part of the chromosome that is deleted, it is associated with the larynx development. So larynx is the voice box in case of human beings. So if the larynx is not developed properly, then the sound production will be affected. So in that case, the individual will have a like as if the mewing of a cat sound will be produced. So that is an example of a deletion mutation. Similarly, there will be a duplication. So as the name indicates, a segment of the gene could be duplicated. So if the sequence of the duplication is same, that is called as a tandem duplication. If the duplicated sequence is attached in a reverse manner, like here it is B and C, but when the duplicated segment is attached, it is C and B. Okay, so this is called reverse tandem. And if the duplicated part is attached at the end terminus of the chromosome, that is called as a terminal tandem. Okay. So an example you can see in the bar eye of Drosophila. So this 16A gene, which is located on the X chromosome of the Drosophila, if it is duplicated, you can see it can interfere with the normal eye development of the fly. So the more will be the duplications, the narrower will be the eye. Okay. So this is what is a chromosomal duplication. So next we have inversion. Okay. So as the name indicates, a segment will be inverted and will be reattached to the chromosome. So like if you look here, the earlier sequences B, C and A, B, C, D and E and F, G, H. But now this B, C, D part has been cut from the chromosome and has been means put here, but in an inverted sequence. So now earlier A and B were closer. Now D has come close to B. So the sequence has been inverted. Okay, like there is a 180-degree rotation is there. Now it could be again two types based on where actually the inversion is happening. If the inversion is happening within the same arm, that is called as a paracentric inversion, which does not include the centromere position. Okay, and the arrangement is occurring within the same arm. Now, sometimes what might occur, the inverted segment might involve the location of the centromere. So, if the centromere is involved in the inversion process, this is called as a pericentric inversion, like you can see here. So, if a pericentric inversion occurs, it can actually alter the shape of the chromosome, like from a kind of submetacentric, the chromosome has become metacentric. Then next we have, okay, this we will come a bit later. First, let's finish the types. So next type we have translocation. Okay. So someone was asking me like whether, okay, Lakshmi Priya and Lalit, you have any question because you have raised your hands? Is there any query from you? Or
No,
it's
ma'am.
like by mistake only? Okay, so translocation means when the segment is exchanged between two different chromosomes. So I hope someone was asking me whether the recombination can occur between a X and Y chromosome. No. If there is a recombination between X and Y chromosome, this will be regarded as a translocation. That is a kind of chromosomal aberration. It is not a means it is not regarded as a recombination. Okay, because this will lead to development of abnormal phenotype. So here there are different types of translocation. See, translocation, trans means what? Different. Location means position. So when the chromosome or a segment of the chromosome is changing the position, we call it translocation. So sometimes the translocation can occur within the same chromosome. Like here, as you can see in the first picture, the chromosome is a segment is shifted from this arm to this arm. Okay. So this is called non-reciprocal intrachromosomal translocation. Intra means within the same chromosome the translocation is happening, and there is means there is it is a one way. Okay, so that is why it's non-reciprocal. Similarly, in the second case, this is a non-reciprocal, but it is a interchromosomal because the transfer is happening between two different chromosomes. Let's say this is chromosome number 7, this is chromosome number 14. Okay. So as two different chromosomes are involved, but there is again a one-way exchange. Only the segment is shifted from 7 to 14 chromosome. So non-reciprocal, but interchromosomal translocation. But sometimes there could be a reciprocal. Okay, like here as you see, this is, let's say, chromosome number 8, this is chromosome number 13 or 14, whatever it is. So a segment is exchanged between them. So the A and B from 8 has shifted to chromosome number 13, and the chromosome number MNO has been shifted from chromosome 13 to chromosome number 8. So this is what is called as a translocation. So broadly, 4 different classes of chromosomal mutations are there. Deletion, a part is deleted. Duplication, a part has been duplicated. Then inversion, a part has been inverted. And then translocation, where a part has been transferred from one location to another location. So is there any question till now?
Ma'am,
No.
Sorry, it's not very clear. Could you please repeat?
ma'am, inversion mutation ଟା ଟିକେ କହିବେ.
Okay, inversion means just look at the diagram. Okay. See, inversion means there is a 180-degree rotation of the segment which has been cut and reinserted into the chromosome. So look at the sequence of the chromosome earlier. What is the sequence? A, B, C, D, E, F, G, and H. So this B, C, and D part, it has been cut from the chromosome, and when it is reattached, the sequence has been inverted into 180 degree. So now what is the new sequence of the chromosome? Instead of A, B, C, D, E, F, G, now the sequence has been A. The B has come to this location and D has shifted to this location. Okay. So now the new sequence is A, D, C, B, E, F, G, and H. And since here the inverted segment is within the same arm of the chromosome, माने सेंट्रोमियर रो किछि पोजीशन चेंज होछि कि होनी सेंट्रोमियर जोठी क्लास 8 अछि सेम आर्म भीतर जेतु रीअरेंजमेंट होछि दिस इज कॉल्ड एज ए पैरासेंट्रिक इन्वर्शन किंतु सेकंड केस रे देखो सेकंड केस रे कोन अछि तमरो सीक्वेंस ए बी सी डी ई एफ जी एच अछि सो जो डी ई एंड एफ अछि ए पार्ट टा इन्वर्ट हिछि सो कोन हिछि ना एफ टा ऊपर को जाइछि डी टा तळ को आछि हला बट ए जो सेगमेंट टा इन्वर्ट हिछि एथरे सेंट्रोमियर प्रेजेंट अछि ओके सो सेट पय के एटा होछि कोन पेरिसेंट्रिक इन्वर्शन
मैडम मझिरे जो गारटा আছে সেটা एक्चुअली सेंट्रोमियर को रिप्रेजेंट करूछि
दिस लोकेशन दिस इज एक्चुअली सेंट्रोमियर सो ए जगह रे देखो सेंट्रोमियर पोजीशन जोटी थिला आगर भी से ई एंड एफ भीतर थिला तो एडी किछि चेंज हेनी परे इन्वर्शन परे भी जेमति थिला क्रोमोसोम सेमति अछि किंतु सेकंड ईयर रे देखो पोजीशन टा कोन चेंज हेछि जदियो ई एंड एफ भीतर अछि बट एडा जेमति तळो को थिला सेंट्रोमियर एबे कोन हेछि सेंट्रोमियर टे ऊपर को उठी हेछि ना So, since the centromere is involved in the inverted segment, this kind of inversion is called as pericentric inversion. Okay, is it clear to all, or
Yes, ma'am.
there is any other query?
Yes, ma'am.
Acha, eta tikke tricky jinsa, so please be a little attentive. Actually, amaro jetale inversion heterozygotes hobo taale kon problem hobo. Okay. So inversion देखिला समस्ते क्लियर अच्छे so अच्छा एडा देखिए मते को तो what kind of a inversion this is whether it is pericentric or paracentric देखो एडा ଗୋଟେ नॉर्मल क्रोमोसोम जोटा अछि ना सीक्वेंस कोन अछि तार ए बी सी डी ई एफ जी एच अछि हां तो को सेगमेंट हमरो चेंज हेछि डी सी बी क्या EFGH ଯେମିତି ଥିଲା ସେମିତି ଅଛି
paracentric
so ଏଟା yes କାରଣ ସେଣ୍ଟ୍ରୋମିୟର ଦେଖ ସେଣ୍ଟ୍ରୋମିୟର ପୁରା ଆଗରେ ଅଛି ଯୋଉଟା କି ଇନଭଲ୍ଭ ହେଇନି ଆମର ଓକେ so this is an example of a paracentric inversion now ଯଦି if there is a person who is heterozygote for this kind of inversion so what does it mean now the person is having a normal chromosome and a inverted chromosome together তাহলে যেতବେଳେ গ্যামেটোজেনেসিস হব so the individual will have a lot of problem so kon pai now when there is a inversion heterozygous or if there is a inversion heterozygous in that condition what can happen na usually amar jetole recombination hobo ba crossing over hebo the means the homologous chromosomes or the homologous region should come side by side but in case of inversion heterozygous, now the B has been shifted to this location, isn't it? Like in the normal chromosome, the B is here, but in the inverted chromosome, the B is far away. But in order for the crossing over to take place, both the B should come side by side. So which is not possible if the chromosome will be linear like this. So in order to avoid this, what the chromosome does, the chromosome forms a loop kind of structure like this. Okay. So now if you see, all the homologous regions are aligned properly. A are together, B are together, C are together, D, E, F. Every region is now aligning properly with their respective homologous region. So up to this part, is it clear why there is a loop?
madam again please
सी ଯେତେବେଳେ ଆମର କ୍ରସିଂ ଓଭର ହଉଛି ନା ତମେ ମନେ ପକା ଆମେ ଯେତେବେଳେ ଦେଖିଲେ ଯୋଉ କ୍ରସ ଓଭର ଯୋଉ A B C ଯାହା ବି ନେଇକି ଦେଖିଲେ ସो ଯୋଉ A ଜିନ୍ ଟା ପାଖରେ A ଜିନ୍ ଥିଲେ ହିଁ ନା କ୍ରସ ଓଭର ହବ
Okay, ma'am.
yes ma'am
ओके ମାନେ A B B एमती साइड बाय साइड रहबा कथा ना तो ए जो लोक जदि यार इन्वर्सन जी की इन्वर्सन हेट्रोजाइगस माने कोन तार गोटे क्रोमोसोम तो नॉर्मल अछि बट आर क्रोमोसोम ता कोन अछि इन्वर्टेड हो जाय छी हला सो ए जगह तुम देखो बी और बी साइड बाय साइड अछंती कि ए जगह टारे बी साइड रे कोन अछि डी अछि
D ଅଛି
सेमती एठे डी पाखो को बी अछि सो दिस इज एक्चुअली प्रॉब्लमेटिक कारण ए जगह रे क्रॉसओवर तमरो हेई परिबनी सो क्रॉसओवर हवा पे कोन दरकार ना सेम रीजन गुडा बा सेम माने जी माने जो क्रोमोसोम रो सेम होमोलोगस रीजन गुडा का अलाइन हवा दरकार सो अलाइनमेंट एमती लिनियरली रहले त अलाइनमेंट हबनी सेत पई ये कोन हेछि ना एमती लूप भले स्ट्रक्चर हे जाय माने मोड़ी हे जाय सो मोड़ी हे जिबा फल रे एबे कोन अछि देखो अलाइनमेंट प्रॉपर्ली हेछि कि नै
Yes.
ए पाखरे ए अछि बी
ଅଛି
पाखरे
ଅଛି
जदि मोड़ी हे गेला अलाइन हे जिबा ना प्रॉपर्ली तो
yes ma'am
a loop kind of structure has been formed. Up to this, is it clear, all of you?
Yes, sir.
yes
Yes,
yes
ma'am.
yes madam
What
Ma'am,
about
this
you,
is
Sneha?
occur during synapses.
Yes, it will occur only during synapses, like during gametogenesis, because only that time the recombination or crossing over will take place. So, Sneha, is it clear to you?
Yes, ma'am. Yes, ma'am.
So now, since let's assume that a crossing over is occurring between the gene B and C. So exactly crossover means what? There will be exchange of parts between the chromosomes like we saw earlier. If this is A, this is B and B. So this part of the chromosome will shift here and this part of the chromosome will come here. So what is the new arrangement that you will get? Capital A, small b. A part ta एठी को जीवो ना और ये B टाइप को पळे आसीबो. So small a, capital B, right? Similarly, if there is a crossing over between B and C, ताहले कौन हबो ना जो एटी क्रोमाटिड रो ना दिया छै टे केयरफुली देखो 1 2 3 और 4 ओके सो एटी क्रॉसिंग ओवर कहा कहा भीतर होछि देखो क्रोमाटिड 1 रे क्रॉस ओवर होउनी
Houni.
क्रोमाटिड 3 रे भी क्रॉसिंग ओवर होउनी ओके सो क्रॉस ओवर रे किए पार्टिसिपेट करछि क्रोमाटिड 2 और क्रोमाटिड 4 हला सो जेताले एमाने पार्टिसिपेट करछन्ते एई जगह रे कोन हबो क्रोमोसोम ब्रेक हबो ओके सो क्रोमोसोम ब्रेक हे कि कोन एई 2 रो रेस्ट ऑफ द पार्ट एठी या संगे अटैच हे जिवो 4 संगे रे सिमिलरली एपटे जहाटा रहिला 2 रो पार्ट सी रेस्ट ऑफ द 4 संगे अटैच हो जिवो ताहले तमर ए भले अरेंजमेंट तयारी होबो ना नहीं गोटे क्रोमोसोम 1 जे कि क्रॉसिंग ओवर रे पार्टिसिपेट बिल्कुल करिनी जेमती अछि सेमती अछि क्रोमोसोम नंबर 4 3 एटा होची एक्चुअली 3 होबा कथा इट नॉट 4 ये भी तो पार्टिसिपेट करिनी क्रॉस ओवर रे सो ये जेमती सीक्वेंस रे थिला सेमती अछि बट जो देटा का क्रोमोसोम पार्टिसिपेट करिथिले क्रोमैटिड पार्टिसिपेट करिथिले क्रॉसिंग ओवर रे सेठी कोन हला ना एठू कट हे गला सो एटा कोन हे गला यार आधा पार्ट जोड़ी हे जाय छि कोटी ए पटे जोड़ी हे जाय छि सो एमती गोटे स्ट्रक्चर तयारी हिची हला पुणी आर स्ट्रक्चर टा कोन हिची ना ए भले एपटे गोटे नुवा स्ट्रक्चर पार्टली क्रोमोसोम 2 पार्टली क्रोमोसोम 4 हला ए भले रिकॉम्बिनेंट क्रोमोसोम तयारी हे जाय छंती अच्छा ए जो क्रोमोसोम टा को देखो जोटा तयारी हिची याद रे सेंट्रोमियर अछि की ए पर्टिकुलर क्रोमोसोम रे there is no centromere so this is what is called as a acentric chromosome so without centromere the chromosome will not survive and it will be degenerated so it will be completely disintegrated अच्छा ये जो तयारी हेची एथरे कोन अछि ना ओटे डाइसेंट्रिक ब्रिज तयारी हेची दीटा सेंट्रोमियर अछि एठी भी ओटे सेंट्रोमियर अछि एठी भी ओटे सेंट्रोमियर अछि तो एमाने जितले सेपरेट हेबे सो रैंडमली ये कोनोसी भी गोटे जगह ब्रेक कर जिवो सो ब्रेक करी गला परे जो तमरो क्रोमोसोम्स तयारी हेले देखो गोटे अछि नॉर्मल क्रोमोसोम सब जीन थिक सीक्वेंस थाई कि गोटे अछि इन्वर्टेड क्रोमोसोम बट तार भी सब जीन सीक्वेंस अछि जदियो इन्वर्टेड सीक्वेंस रे अछि बट ऑल द जीन्स आर प्रेजेंट बट ए जो दीटा नुवा रिकॉम्बिनेंट क्रोमोसोम तयारी हेले देखो कोन अछि ना आधरे अधा जीन्स तमरो नाही बा अधा क्रोमोसोम रो सेगमेंट्स नाही सो ए जो दीटा क्रोमोसोम हेबे दे विल डीजेनरेट ता माने तमरो जदि चारिटा प्रकार गैमेट तयारी होची चारिटा गैमेट तयारी होची ता देरु 50% ऑफ द गैमेट्स विल बी इनवायबल so जो individual माने or those individual who are actually inversion heterozygote they will have a problem during the gametogenesis or they will have a reduced fertility because 50% of the gametes produced by them will be inviable understood ei jinsara बुझ पारलो समझते
मैम आउथरे टिके सेकंड
Inviable माने मैडम बुझता
inviable माने they will not survive yeah.
Okay.
Like for example if this is a human like male so from if in one meiosis among the 4 spermatozoa produced 2 sperms will be means killed or they will be completely disintegrated yeah. or they will be lost only 2 sperms will be there so the viability decreases by 50%.
Okay, ma'am.
So someone else was asking something.
No, ma'am, understood.
Understood. Okay, fine. So similarly, this is in case of your paracentric inversion. So in case of pericentric inversion also, you will have a similar problem like that. But only the type of chromosome produced will be little bit different. So as here in case of pericentric inversion, you had a dicentric chromosome and an acentric chromosome. But in case of pericentric inversion, you will have chromosomes with 2 chromosomes will be normal, viable, with a normal sequence and an inverted sequence, but 2 chromosomes will be there, which will be produced with multiple gene deletions and duplications. So here also the viability will decrease by 50%. Okay. Then coming to translocation. So in translocation, heterozygotes also similar kind of problem will occur during the meiosis. The in one case, like depending upon how the chromosomes are being segregated, so either you will have this kind of normal gamete where although the segments will have translocated genes but all the genes will be present but in rest of the 2 cases depending upon how the chromosomes are being segregated you will have deleted and duplicated chromosomes and again these kind of gametes will be inviable. Okay, so you just go through this is easy to understand. So the whole point of this discussion is what? Now, if there is inversion or there is translocation, heterozygote, zygosity in any individual, then it will reduce their fertility by 50%, because the gametes produced in case of inversion or translocation heterozygotes, like few of the, or like most of the gametes produced by them will not be viable. Okay, so that is actually a problematic condition. So is there any question from any of you till now, or is it clear to all of you?
मैम टिके लास्ट टेस्ट कहले
This translocation one?
ट्रांसलोकेशन हाँ
Okay.
ट्रांसलोकेशन रो लास्ट स्टेज टा
Wait. See here. Okay, let me explain it also. So here if you see translocation heterozygote, whatever it is, okay. See, the first individual, it is having normal chromosomes, right? N1,
Okay.
N2, so all the genes are normal. But in the second case, you have translocated chromosomes, because like from color combination also you can know that a part of this chromosome has shifted to this one and a part of the green has shifted to this one. So there is a hybrid kind of chromosome is there. So this is a translocated chromosome. Both of them are translocated chromosome. Now during meiosis, as we saw in case of inversion, the proper alignment should be there so that the genes could come in close to each other. Okay, so this is how the chromosomes will be aligned, or the chromatids will be aligned during the meiosis. So these are the normal chromosomes. Okay, these are the translocated chromosomes. Now from this point onwards, there are three ways by which the separation of the chromatids can occur during the cell division. So what are the possible separation? Now one is alternate. Alternate means what? Now this chromosome will move to one pole and these chromosomes will move to the other pole. Okay, this is what is called alternate. So look into the next slide. The first picture, that is an alternate segregation. So in the alternate segregation, what you have? You have both the normal chromosomes in one pole and you have both the translocated chromosomes in one pole. But that is fine, because even though the translocation is there, both the like the entire set of genes are present. So the gametes will survive. Okay. But look at the other cases. The other two cases, if you observe, here what could be another way of translocation? Sorry, another way of separation. Another way of separation could be like this, vertical separation. So one pole will have this chromosome, other pole will have these chromosomes. So here if you see, this part is duplicated or not?
Yes.
And the other part is missing, half of the orange part is missing. Here the orange is duplicated but half of the green part is missing, right? So both are having abnormal chromosome. Similarly, another possibility of segregation could be like this, horizontal segregation. Like one pole will have this chromosome, other pole will have this chromosome. So here also, see, the orange part is duplicated, the green part is missing. Here the green part is duplicated, the orange part is missing. Okay. So in both these cases, you will have abnormal set of chromosomes. So that is why the gametes will not be viable. So that is what is explained here. Adjacent one, adjacent two, like horizontal and the vertical segregation, you will have non-inviable gametes. Only in the alternate segregation, you will have viable gametes. So is it clear to all of you?
Yes, ma'am.
Yes, ma'am.
Okay, fine. So since it has already passed the time, let's stop here. I will try to compensate it in the next class only. Now, if anyone has any personal questions, you can ask. Like if you have your individual queries, now you can ask. Or else we
Hello,
can.
ma'am. Hello.
Yes.
Hello, ma'am. Previous class रो और today class रो PPT टिके send करबे.
हाँ
Group
ठीक
रे.
अच्छे मैं आज ही शेयर कर दूँ. Okay.
Ma'am आपको number कहले हमने message करिवो. ना आपन group
Actually
रे अछन.
ମୁଁ ଜାଣିନି whether it is allowed or not, ମାନେ ଏଟା କାଉନସିଲରଙ୍କ ସହିତ ଇଏ ମାନେ ମୁଁ ଜାଣିନି ଏଟା ମାନେ
ପୂର୍ବରୁ ଯୋଉ
ଏଟା ତ ପରମିସିବୁଲ କି କଣ, ମୁଁ ଏଟା ଗ୍ରୁପରେ ତା ମାନେ ଇଗ୍ନୋର ଅଫିସିଆଲ ମାନେ ଅଫିସିଆଲ ମାନଙ୍କ ସହିତ ସେୟାର କରିଦେବି, ସେ ତମ ସହିତ ମିଶିବ। ତ ମୁଁ ନଟ୍ ସିଓର କି ଏଟା କେତେ ଦୂର ଆଲାଉ।
Ma'am, excuse me.
Yes,
hello
yes,
ma'am.
Ma'am,
Hello.
I am having one query.
yes, you can ask.
Yes, ma'am. Actually, I am not connected in any official group where I can get the link of the classes. Like I have to depend on the other person to get the link for the classes. So from where can we get the link? That which class is
Actually,
going to?
I have no idea. Actually, we counselors have been also added to one group from where we are getting all the information. So for this,
मैम मेल को जाइए मेसेज जाइए मेल को.
Okay, I'm not getting actually the mails. I'll
no,
see
you
to
can
that
check
thing.
in your spam folder or task folder. You can
Okay,
check.
okay, okay,
Maybe
okay.
it will be
I'll see
there.
to that thing. I'll
Yes,
check that thing. Thank you.
yes. You can just have a look at there.
Okay, okay, I'll do that.
Okay.
Thank you.
Any other
He-
question
he-
from anyone?
hello ma'am.
Yes, Vidyasagar.
ମ୍ୟାମ୍ ପ୍ରିଭିୟସ୍ କ୍ଲାସ୍ରେ ସାର୍ ଆମକୁ ସାର୍ଙ୍କ ନମ୍ବର ଦେଇଥିଲେ ଆଉ ଆମେ ମେସେଜ କରିଥିଲୁ, ସାର୍ ପିପିଟି ସେଣ୍ଡ କରିଥିଲେ।
ଓକେ ଫାଇନ୍, ବଟ୍ ମୁଁ ଜାଣିନି ଆକ୍ଚୁଆଲି କେତେ ଦୂର ଏଇଟା ପରମିସିବଲ୍। ମୁଁ ଗୋଟେ ଥର ଜଷ୍ଟ ଖାଲି ପରମିସନ୍ ନେଇଯାଏ, ଇଫ୍ ଇଟ୍ ଇଜ୍ ଦେର୍ ଇଜ୍ ନୋ ପ୍ରୋବ୍ଲେମ୍ ଆଇ ୱିଲ୍ ଅଲସୋ ସେୟାର। କିମ୍ବା ୟୁ କ୍ୟାନ୍ ଆସ୍କ ଲାଇକ୍ ୟୁର ଅଫିସ୍ ପିପଲ୍ ଅଲସୋ ଟୁ ସେୟାର ଦ ନମ୍ବର ଅଫ୍ ଦ କାଉନ୍ସିଲର, ସୋ ଦେ କ୍ୟାନ୍ ଅଲସୋ ଡୁ ଦ୍ୟାଟ୍। ଆକ୍ଚୁଆଲି ଜଷ୍ଟ ମୋତେ ଗୋଟେ ଥର କନଫର୍ମ ହେଇଯିବାକୁ ଦିଅନ୍ତୁ, ମାନେ ମୁଁ ଜାଣିନି ଯଦି କରିଦେଲୁ ପୁଣି ପରେ କିଛି ଯଦି କହିଲେ ଦ୍ୟାଟ୍ କୁଡ୍ ବି ଇସ୍ୟୁ। So ମୁଁ permission ନେଇଯାଏ, by the end of the session I will share it. Okay, no problem. There is no
ଓକେ ମ୍ୟାମ୍।
problem from my side. Okay, so any other question from anyone?
No, ma'am.
ମ୍ୟାମ୍ ଗୋଟେ ଡାଉଟ୍ ଥିଲା, ୱାନ୍ ଜିନ୍ ୱାନ୍ ଏନ୍ଜାଇମ୍ ହାଇପୋଥେସିସ୍ ଟା ଟିକେ ବୁଝେଇଦେବେ?
अच्छा, see one gene one enzyme hypothesis ର concept होची कि usually ଆମର जदि आमे देखिबा कि there are suppose in a metabolic pathway multiple enzymes are involved, right? So like if it is like कि जदि गोटे जीन रो जदि multiple polypeptide बा multiple enzyme synthesis हो थांता ताहले कौन हुआ, even if को भी segment of gene जदि non functional ही गला, the pathway टा inhibit होबा कथा नुहे, it should be compensated by the same gene। जेहेतु गोटे जीन मल्टीपल प्रोटीन बा मल्टीपल एंजाइम सिंथेसिस करि पारुची, सो से प्रोसेस रे कोनोसी प्रकार माने माने कोन कहि पर माने कोनोसी प्रकार इंटरप्शन होबा कथा नुहे। बट तुम देखिबो कोन ना जदि गोटे मेटाबॉलिक पाथवे रे बा प्रोसेस रे तुम कोनोसी गोटे जीन को माने डैमेज करछ बा यू आर मेकिंग ए जीन नॉन फंक्शनल, ताले एंटायर पाथवे विल बी इनहिबिटेड। जहार माने जो थ्रू हमको कौन ये मिलूची कि एक्चुअली गोटे एंजाइम इज एनकोडेड बाय दैट पर्टिकुलर जीन, जो कि जदि गोटे जीन रो फंक्शन को तुम डिसरप्ट कर दोच, दैट एंजाइम इज गेटिंग डिसरप्टेड एंड accordingly the entire metabolic pathway is being interrupted। Yeah, so that is the concept of one gene and one enzyme or one gene one polypeptide hypothesis।
Okay, ma'am. Thank you.
Okay, what about others? Is there any other query from anyone?
No, ma'am.
Okay, fine. See, I am trying my best, किंतु honestly कहिया गले तुम genetics रो माने बाकी subject मु जानिनी, but this topic is very vast। तुम देखी परियो बहुत सारा syllabus अछि, तो एई पेस रे जिबा जदि हम केबे भी syllabus complete तो कला करिबा difficult लागुची। Anyways, I will try my best। So मु समस्त को request करबी कि जदि तुम पाखे materials already अछि, तो गोटे थरो टिके reading करी के आस्तिले easy होबा टिके बुझिबा पई कि आउ इवन कोरिलेट भी करिया को तुम मानक टिके माने कंपैरेटिवली इजी होबो जिंसटा पूरा समथिंग जदि नुआ पढ़िला को तो जहाँ भी टिके डिफिकल्ट हम, से बेसिक क्वेश्चन भी अराइज करिवो। सो जदि प्रीवियसली गोटे थर निज तरफ टिके पढ़ी के आसथिबो ताले तुमको कोरिलेट करिया को इजी भी होबो, आ इवन क्वेश्चन गुडा ऑलरेडी तुमरो फ्रेम हे थिबो, आ जो गुडा क्वेश्चन जहाँ फ्रेम हे थिबो मु डिस्कस कला बेले सेइथरे गुडा जिंस सॉल्व हे जिवो। इंडिविजुअल एते गुडा क्वेरी सॉल्व करिया मोर किछि प्रॉब्लम नाही, बट प्रॉब्लम होची तुम टाइम बहुत लिमिटेड अछि। So if we keep on solving the queries only, we can never complete the syllabus. That is the problem.
Okay, ma'am.
So if there is no more question, we can wind up the class. And regarding that sharing of the number, I will inform you tomorrow. Okay. So if there is no problem, I will share my number tomorrow, and we can stay connected in the group.
Thank you, ma'am.
Okay. Okay then. Thank you
Thank
so
you.
much.
Thank you, ma'am.